officially commencing the workshop, that is the micrometry in microscopic image analysis using ImageJ software. So this is going to be the schedule for today, and we are going to have both lectures and live demonstration which are conducted via Zoom. And I hope that this workshop will cater all the queries related to image analysis using the ImageJ software and help you in pursuing the same in your corresponding institutes. Herewith, we are introducing our speakers and chairpersons for the day, and we are extremely grateful to the leading and eminent anatomists all over the country who have agreed to be the chairpersons and speakers for today's session, and this is our great pleasure in hosting them for our workshop. We were planning to have the workshop as an offline session so that the participants could come directly to our department and have a hands-on workshop using ImageJ analysis software and do and process the slides. Initially, the idea started with the 15 to 20 participants, but due to the ongoing Omicron pandemic, we could not pursue the offline form of the workshop. Following it, we had conducted and we have planned in the form of an online workshop in the Zoom, and beyond our expectation, that is the real words which I have to use, much more than our expectation, the audience response was huge right from the first day. So even though I cannot welcome you directly to the beautiful place of Puducherry, I am welcoming you in online mode to the beautiful place of Puducherry, where our institute is located. And I hope most of you, if not all, would have visited Pondicherry once or more than once, and you would have very good memories associated with you for this city. So next time when we are hosting the workshop, we will try to plan it in a offline mode so that you can visit and also you can enjoy the city directly. Our institute, Jawaharlal Institute of Postgraduate Medical Education and Research, has a profound and a long history associated with it. It started as L'École Pondicherry long back, and following it, it has undergone various stages of metamorphosis. The last and the final metamorphosis was when it became the Institute of National Importance, and following it has spread its wings in various dimensions. Today, the average OPD census of the institute ranges about 2,852 with more than 1,000 inpatient admissions every day. We have various courses including MBBS, MScs, PhDs, and also postgraduate in anatomy, and we are conducting workshops and also the CMEs in different sectors of anatomy. In the institute, there is located the JIPMER Academic Center, and in the JIPMER Academic Center, it is especially dedicated for the anatomy, physiology, biochemistry, and medical education. In the ground floor of JIPMER Academic Center, is the Department of Anatomy. So again, I welcome you virtually to the Department of Anatomy. And in the Department of Anatomy, we are basically working upon histology, embalming fluids, surgical skills workshop, and radiological anatomy, in addition to the other routine realms of the anatomy. So with this introduction, I welcome you all to the online CME on microscopic image analysis using ImageJ software. As all of you know today, the histological studies constitute one of the important fields in the research of anatomy. While we are using the histology, for a long time we have been using the qualitative methods for analyzing. Of late, we have the softwares for the image analysis and also for morphometric measurement. Out of the various softwares which are available, which could be used in the microscopic image analysis, ImageJ software is one of the highly used one because of its user friendliness and also because of the freely available status of it. I am not revealing much details about the microscopic image analysis and also the ImageJ software because I am reserving it for the first presentation, that is the introduction part, which will be delivered shortly. So I hope you have a wonderful academic feast today, and at the same time, the introduction to the ImageJ analysis will help you in improving the research province which you have been doing related to the histology till now. With this introduction, I am now introducing our CME coordinator, Dr. Suman Verma. Dr. Suman Verma has the qualification of MBBS and MD Anatomy, and she is currently additional professor in the Department of Anatomy, which is located in the Academic Center, JIPMER. And now, Madam will be introducing the chairpersons for the session. And at the same time, it's time for me to sign off at the moment. And I am Dr. Dinesh Kumar, currently Associate Professor in the Department of Anatomy, Academic Center, JIPMER. So to begin with, first we are going to have the lecture on Introduction to the ImageJ software. And this lecture will be delivered by Dr. Suma HY. So to begin with, we are going to have the lecture Introduction to ImageJ software, and this lecture will be delivered by Dr. Suma HY, Professor and Head, Department of Anatomy, JIPMER. Dr. Suma HY has the academic qualification of MBBS and MD Anatomy and currently Professor and Head in the Department of Anatomy, JIPMER. Madam has done her undergraduation from Sri Devaraj Urs Medical College, Kolar, Karnataka, and her post-graduation degree, that is MD Anatomy, from MS Ramaya Medical College, Bengaluru, Karnataka. Madam has more than 15 years of teaching experience, which includes both undergraduate and postgraduate. In the teaching experience, Madam has guided many postgraduates during the particular tenure. Her fields of interest are histology, DNA damage studies using comets analysis, and gross anatomy, in addition to the museum techniques, including the corrosion cast. Madam has more than 20 publications on her name in the national and international journals. Madam has authored a book along with the colleagues of JIPMER, which includes the anatomy and physiology for the nursing students. And Madam has presented considerable number of papers in both state and the national conferences. Now, I call upon Dr. Suma ma'am to give her talk. Thank you.
Thank you, Dr. Dinesh, for that wonderful introduction. So, good morning to all the delegates, the chairpersons, my department faculty, and the residents. I welcome you all to the National CME on micrometry in microscopic image analysis using ImageJ software. In 2018, we had organized a workshop on micrometry, similar workshop in a offline mode, wherein we had participants from nearby Puducherry as well as from the outside states. And we had about 45 participants, and it was highly appreciated. So we thought like again we can conduct this year, but because of the one more wave of COVID, we had to postpone it, and we were just thinking like how to organize it, and we thought like we can go for a online mode, even though offline is a better option always, but because of constraints we couldn't conduct it. Now once we released the brochure, we had a huge response from the day one, and it increased enormously till yesterday and day before it crossed 1,000 registrations. So I am very happy that we have got such a great response from the faculty, the residents from all over India as well as abroad. So I like to share my screen now. Is the screen visible? Dr. Dinesh?
Yes, ma'am, it's visible.
It's visible. Okay.
Yes, ma'am.
So it's my pleasure to introduce all the delegates to the ImageJ software. Now, first of all, what is an image? So image means like we have usually seen in our histology slides, image like this. So this is a Purkinje neuron, which has a cell body as well as the cell dendrite. So this is what. So this one is a 2D image, which actually visualized in a 3-dimensional picture. It will have a cell body and the dendritic branches which are spread in various directions. So this is a comparison between the projection of a 3D image and a 2D plane image. So it's just like you cut across this 3-dimensional cell, and at one particular point, as if on a paper, the image has been represented. So this is what we are visualizing in the histology slide. But it will have a dimension in a 3-dimensional mode. So this we can interpret by considering this, comparing these two images. So here what you can see is a 3D image. It will have a 3 axes of coordinates in a spatial representation. When we represent spatially, 3 axes can be is said to be there. One is a x-axis, one is a y-axis, and one is a z-axis. So that's a 3D image. So for a human eye, we are unable to visualize in a 3D image in our routine work. So what we generally visualize is the image in a x-axis and a y-axis. So it is said to be in a 2D plane. Now, what is a digital image? A digital image is just a matrix containing numbers. So, what you see here, this is a eagle. When you magnify it, you will see that it is made up of a matrix of different shades, and each shade, now you can see inside that small boxes are there. So this we call it as a one picture element within this matrix. Each of these tiny units, they are called as a one pixel element. Pixel element in the sense pixel. So pixel means it's a picture element. A single cell of that matrix is called as a pixel. So pixel, actually it will be very small, and we cannot see with our naked eyes. And because of this, as the number of pixels increase, the image appears more smooth, and the clarity also will be more. And in a digital image, each of this unit, it will have a value depending on the bit within the each image. Now, the digital image, the advantage is of a faster processing. It is responsible for efficient storage, as well as allows for a image manipulation. And the disadvantages will be like, even though the quality of the image is very good, it requires a higher memory, and hence the system where we work, it needs to have a faster processor. So we just understood what is a digital image. Now we'll go to image processing. So being in this field, as we are coming across the images, there is a need to process the images. So what is the image processing? It involves converting an image into fundamental components to extract meaningful information. So whatever the image we are having, we have to So image processing involves converting an image into fundamental components to extract meaningful information. So just like in our routine daily work, we come across various type of images, like a painting, or like when we see any picture. Whereas in context of the digital images, digital images are samples of information. So these are sampled at vertex points of n-dimensional grids. So example of image processing is like, we can find the shape of the particular area of interest, detecting the edges, removing noise, and counting the objects, etc. Now image processing, it covers a different range of techniques. So I am going to mention 4 such techniques. One is a image enhancement. Image enhancement, it's a procedure of improving the quality and information content of the original data before processing. So before we start processing, we need to work on making it a more presentable for display, as well as it is fit for analyzing. So that is improving the quality and the information content of the original data. This includes contrast enhancement. For example, if you see here, in the first image, you will see that the contrast is not much made out. But once we apply some filters, the contrast is much well made out. So this is the image enhancement. So it includes contrast enhancement using histogram, spatial filtering, density slicing, brighten an image so that we are able to identify the key features. Now the images are adjusted so that they are more suitable for further analysis. Now coming to the second technique, image segmentation. So what is image segmentation? It is nothing but partitioning a image. So you can see an image here. This image, we will partition it into multiple components like this. So this involves so this image segment can involve separating a foreground from a background. For example, in image, we have some images which are like when picturized, the image, the components which are in the front, they are much well seen compared to some of the components which are in the background. So to analyze whichever one we want, we need to separate that. Second type is clustering the regions of pixels based on similarity in color or a shape. So we can separate them based on the pixel, like if a one area is there, so surrounding that with the similar pixels, it can be separated and studied. So this is useful whenever we want to, like in a 3D MRI scan, to separate a tumor tissue from the adjacent normal tissue. And the next is the third technique is noise. So the noise is a result of errors in image acquisition process. So when we are acquiring an image, there's a possibility that without our knowledge, some noise will be introduced. So this will affect the pixel values, but this does not reflect the true intensities of the real scene. So there are some filters which are available which will help to remove the noise. So if you see in this picture, you can see that this type of a noise is called as a salt and pepper. So you can see in the background salt and pepper appearance, and using a vernier filter, this has been removed. Now you are able to, the object of interest is a more clearer. And the fourth technique is the analyzing region of interest. So if we have an image like this, and if we want to demarcate the region which we want to study, so we can demarcate it by marking the edges like this, and we can measure the length, the area of this. So that is called the region of interest. So like this, four techniques will be able to go about our image processing. Now we understood what is a digital image, how to go about the image processing. Now about the image J. So in this picture, you are seeing a scientist called Mr. Wayne Rasband. He worked in National Institute of Health, USA for about 40 years, and is a software developer. He used to develop software for biomedical researchers working with the microscopic images. And initially it was not named as an image J. In 1987, Mr. Wayne, he started writing software for Apple Macintosh for image analysis, and he named it as NIH. But the problem later, it was observed that it couldn't be used with the Windows system. Then later in 1995, the Sun Microsystem developed Java programming language. So after which Mr. Wayne ported NIH to Windows and maintained a single multi-point version of the source code. So after which it was called as a image J. In the sense, J means Java. So because it is based on Java runtime environment, it was labeled as a image J. Now even after retirement, he continues to work as a volunteer, wherein he fixes bugs, answers questions, and adds the any newer developments based on the feedback from the users. And he is also involved in maintaining the image J website and the mailing list. Now coming to image J, it's a Java based software used in image processing, and it can be run on any system with a Java runtime environment, and it works across either a Windows, a Mac, or a Linux. And the specialty of this is the user friendliness can be extended with the use of what is called as a macros and the plugins. Now, Mr. Wayne made sure that it was kept as an open source. So this allowed for many researchers and scientists who are involved in the image analysis to add a new features to the software. So we have active collaborators worldwide who help to improve and maintain the versatility of the software. Then the two main advantages of this is a plugin and the macros. So what is a plugin? Plugin is nothing but a small computer program which will be utilized for a particular task. Now, most of the image built-in menu commands, what you will be learning later, are in fact implemented as a plugins. Then what is a macros? Macros are similar to this one, but these are small programs, computer programs, which will contain the sequence of operations, which will perform when analyzing. So, as a scientist is analyzing a image, what all the steps which will be done, which can be noted and developed as a one file which can be stored, and this will be added to the software. So subsequently, similar images can be analyzed more smoothly following the same steps. So a lot of macros are there depending on the nature of the image analysis by a particular person. They can download those macros and use for their analysis. Now, what is ImageJ for? So there are a vast number of papers. If you go through the literature, there are a vast number of papers who have used the ImageJ software, which spans across so many disciplines, including medical science, the material science, etc. So there are papers from confocal microscopy to X-ray analysis, vehicle license plate detection, ultrasound diagnosis of breast cancer, development of automatic 4D segmentation algorithms, and CT image reconstruction. So it depends on how we explore it and use to analyze the image. So we know like what are its applications, but what it is not for. It is not a replacement for Adobe Photoshop or any other graphic photo editing software. So generally the commonly used everywhere for our regular photos is Adobe Photoshop, but the purpose of Adobe Photoshop is totally different, whereas the purpose of the how this image has gone about is totally different. It is meant for quantification, filtering, measuring, and mathematical processing. Then the scientist Richard Stallman, he has defined certain freedoms in ImageJ software. So the first is the freedom to run the program for any purpose. Second is the freedom to study how the program is running and ability to make changes to improve the program. The freedom to redistribute so that any newer development is utilized and shared with the scientists for improving the analysis. And lastly, the any improvement which has been brought about by the researchers to be freely shared with the community and for the dissemination of the knowledge. So these are certain advantages. So it has the most commonly used because of this. Now coming to what all the things we can do using ImageJ. So the ImageJ supports raster image formats like TIFF, GIF, JPEG, raw data files, DICOM images, FITS, BMP, PNG, etc. So you can see it is so versatile. So it supports almost all the image formats. It also can stack series of images which share a single window. It can calculate area and pixel value statistics for user-defined selections. and we can measure distances and angles. Now, out of these image formats, TIFF is the most preferred format to work in imaging, since it can store the non-image properties as metadata. So, in case of if you convert any digital image to a JPEG format, some of the information in the picture will be lost. As a result, TIFF is the most preferred format, where the DICOM is the standard for medical imaging. Now, the software can display, edit, analyze, process, and save colored as well as the grayscale images from 8-bit to 32-bit. So, this is what you'll be going to learn how to do all these functions using the software. Then you can do geometric transformations like scaling, rotation, and flips. Then standard image processing functions like contrast manipulation, convolution, Fourier analysis, smoothing, sharpening, error detection, and median filtering. You also can create a density histogram and line profile plots. Now, in this image, you can see this MRI scan using this edge detection tool. Only the edges are delineated. So, lot of options are available. Only thing researcher needs to explore all of this. So, to summarize, why image processing? So, the first point is to improve the appearance of the image, and second is to carry out the quantitative measurements. Now, finally, why we need to do the image processing? Importance. So, in your routine work, the analysis of histological images is a subjective component depending on the expertise of the observer. So, what are the analysis we do? Like focus on the tissue structure, distribution of the cells in the tissue, regularities of the cell, etc. So, all these observations are subjective, and this is a time consuming and prone to intra-observer and inter-observer variability. Hence, there is a need for a computer-assisted image processing to quantify the histological structure. So, it is a more objective assessment. And it has been observed that the scientific articles which contain a quantified information whenever a histological analysis done has a higher chance of getting published. Then if we see in the pathology, it aids in accurate diagnosis of pathological conditions, for example, grading of cancer. It is also useful in automation of diagnostics to handle higher patient load. So, if some diagnosis has to be done, if it's automated, the subsequent slides can be easily fed to the system, and it will give the data which can be analyzed, and the report can be given to the patient. In the field of research, to understand the biological mechanisms of disease process, and also example like quantifying expression of level of proteins in different compartments of a tissue, which forms the basis for biomarker discovery. For example, in this picture, you are seeing here a HND stain slide, whereas here using the ISC technique for the estrogen receptor. So, the distribution of the estrogen receptor within the different tissue components has been made out. So, using we come to know how the these biomarkers can be more utilized for arriving at a discovery and for the patient care. So, this is the 2 slides on the importance of image processing. So, for the anatomy, it's mainly like quantifying the histology image, whereas in case of a pathology, it's more of a applying to the patient care grading the different stages of the disease, as well as the biological mechanisms and biomarker discovery. So with these few slides, I have tried to cover the what is the image processing, how the image J came into being, and what are the applications and how the software can be explored for various user-defined applications. These are my references. So with this, I thank all the speakers who agreed to give their talk for this CME, as well as the eminent chairpersons who have agreed to share their time with us and give their valuable input. So, and I also wish to thank the organizing team of our department who have taken great effort in working very hard from the day one, like from the registration, the IT development, how to go about organizing, how to manage the delegates, then answering all your queries. So they have put a lot of effort, and I am very much hopeful that it will be a great success. Thank you.
Thank you for the wonderful session, ma'am.
Thank you, Dr. Dinesh.
Okay. So by this session, we got to know about what all the benefits of doing image processing, and especially in today's era where the journals seek a better quality images, image software will be of immense use in doing the image processing and the post processing of the images to get better quality images, which can be used for publications, and at the same time the measurements also can be done usefully. And it was the session was crisp, and it addressed it indulged the curiosity of the audience for the further coming sessions. So I would like the audience to ask questions, and if you have questions, it is very difficult for us to unmute individually and then attend the questions. So I would suggest that the audience can post their questions in the chat box as and when they have a question, and at the end of the session we will be pulling the questions and we will be presenting to the speaker so that the speaker can answer those questions. If you have any questions regarding the introduction to the image software and image processing, you can post in the chat box, which we will take up for the answering. Okay. Okay. Suma ma'am, one question has been asked: how to quantify histology images, whether it is tissue specific or it's general?
It will be taught in the future session. That is what is the last session is on. How to measure the.
Then one more question is how to remove noise during processing.
Okay. So in the software, there will be one option like this is what user interface. When we take you to the user interface session, there will be showing you all the options by which this noise removal can be done, and how to do the measurement. It will be taught, and there will be option to do the statistics. Once you take a measurement, there is one option for the statistics. So all this, it will be coming up in the future sessions.
Okay. Ma'am, another one question: is this software updated for counting specific number of cells, example ligamentocyte, or it is manual?
Even the quantification of the numbers also can be done. Once you define the particular object, it can be made to apply to the similar objects, and there's a count option. like counting a nuclei, counting a cell, like that, it can be done. It will be taught. It will be taught in the future session.
Another one question is it free software? For that, the answer is yes. The installation of the software will be taught in the upcoming session.
Yes.
Then one more question is, what is stack series of image sharing a single window?
Yeah, stack series means it is a confocal image. Confocal image, it takes a section in a focal length, like it's a three-dimensional. So across that particular object, it takes a series of section, and we can upload that in the image software and analyze that also. So that stacks option you will be able to see once you go to that menu within the image. There is one option where how to analyze the stack. It will be shown to you in the user interface.
Yes, ma'am. Can we use image J for gross specimen and bones?
Yes, it can be used.
Yes, ma'am. Yes, ma'am. So, is this software used for volume measurement of the brain structure, MRI based images?
Yes, madam. It can be used also.
Yes, ma'am. So with this, we are concluding for the cost of the software, it is freely available, and the installation of this software will be taught in the upcoming session. So with this, we were concluding the first session, and I again thank Dr. Suma ma'am for the wonderful introduction to the image processing and the image software. And we hope the other questions will be answered as and while we will continue the further coming sessions. Okay. So
thank you, Dr.
Yes, ma'am. So I would request Dr. Sharif sir to unveil. We will be unveiling the virtual certificate of Dr. Suma ma'am for rendering this session, and I would like Dr. Sharif sir to virtually hand over the certificate to Dr. Suma ma'am.
Good morning, Dr. Suma. Thank you. Yeah.
Good morning, sir. Thank you, sir.
Yes, sir. So here we are unveiling the certificate for Dr. Suma ma'am for the presentation. Yes. Thank you, ma'am.
Thank you.
With this,
thank you.
Yes, ma'am. So with this, we are going to the next session of the day. The next topic is again going to be a lecture, micrometry and its application in medical research. This session will be lectured by Dr. Vasudha Kulkarni, Associate Professor, Department of Anatomy, Dr. B.R. Ambedkar Medical College, Bengaluru, Karnataka. Dr. Vasudha Kulkarni ma'am is MBBS MD Anatomy graduate. Madam is currently working as an Associate Professor in the Department of Anatomy, and she is a convener in the Institutional Medical Educational Unit, Dr. B.R. Ambedkar Medical College, Kadugondahalli, Bengaluru. Madam had her graduation from MBBS graduation from Sri Devaraj Ars Medical College, Kolar, and post-graduation MD Anatomy from Dr. B.R. Ambedkar Medical College in the year 2010. Madam has a fellowship in the FAMER, that is the Foundation of Advancement in Medical Education Research from CMC Ludhiana and 2012 and 2014 batch, and NMC recognized advanced course in medical education from St. John's Medical College, Bengaluru in the year 2015-2016. The chairpersons for this session will be introduced by Dr. Suman Verma. Ma'am, it's over to you.
Thank you, Dr. Dinesh. We have our esteemed chairpersons with us, and it's an honor to introduce such senior faculty. First, we have as chairperson of this session, Dr. Sharif A. Sir is MBBS MD Anatomy, Professor and Head Department of Anatomy, Professor in charge Computer Facility, Professor in charge Telemedicine Facility, Founder Professor in charge Clinical Ecotoxicology Facility at All India Institute of Medical Sciences, New Delhi. Sir has done UG and PG from JIPMER, Puducherry, and has work experience of more than 34 years. guided many postgraduates during the tenure, and Sir has 6 ongoing research projects. Sir's field of interests are clinical ecotoxicology, heavy metal toxicity, enteric nervous system, and developmental biology. We welcome you, sir, to the session.
Thank you, everybody.
Thank you, sir.
Yeah. I would like to first thank the organizers of this CME. And it's a pleasure to be associated with a CME being organized by my own very own alma mater. And my IT journey actually started from JIPMER in the year 1982 or something. It's about 4 decades ago. We used to have a professor, Dr. Sethuraman, who started a doctor's computer club. And that is where my my information technology journey started. And today I am heading the IT division of the All India Institute of Medical Sciences. It's a pleasure to be associated with you. Thank you for the kind words and a nice introduction. Thank you.
Thank you, sir. It's an honor to have you here. Our next chairperson is Dr. Daisy Sahani, ma'am, Professor and Head, Department of Anatomy, Postgraduate Institute of Medical Education and Research, Chandigarh. Ma'am is President, North Chapter of Anatomist Society, Fellow of National Academy of Medical Sciences, Member Editorial Board in Clinical Anatomy published by Mayo Clinic, and Translational Research in Anatomy. Also member of National Academy of Medical Sciences, India, life member of International Association for the Craniofacial Identification, life member of International Society for Heart Research of Indian Section, life member of Indian Science Congress Association. Ma'am has published more than 150 original articles in journals of repute and has many awards to her credit. We welcome you, ma'am, to the session. Welcome, ma'am. Now I hand over to
Namaskar.
Namaskar, ma'am. Welcome. Welcome.
Thank you. Thank you very much for your kind words. Thanks a lot. And I wish you great success to the CME. And first lecture was excellent. And I hope next lecture will be very good. And I wish you good luck, all of you. And thanks for inviting me.
Thank you so much. Over to Dr. Dinesh.
Now we will be having the session. We welcome Dr. Vasudha Kulkarni ma'am to conduct the session.
Dinesh. Dr. Dinesh. Hello. Dr. Dinesh.
Dr. Vasudha, you're audible.
Yeah. Can you help me to share the screen? I'm not able to share my screen.
You can click downside there, share button is there.
No, disabled participant screen sharing.
Okay, give me 1 second.
Yeah.
No, but you are co-host, ma'am. You are
Host
okay.
disabled.
One second.
Yeah, I think now I'm able to do it.
Ma'am, you can share now. You can share.
Yes. I could share it. Thank you. Good morning to respected chairpersons, delegates. To begin with, I would like to express my humble and sincere gratitude to
Dr. Suma, Professor and Head of Department of Anatomy, and her organizing team for giving me this opportunity to share few of my thoughts and views about micrometry and its application in medical research. Now, we are familiar with Antony von Leeuwenhoek, the father of microscopy, who had not only invented microscope, but had done earlier most method of measuring the cells. So he had used fine grains of sand to measure the size of erythrocytes. Since then, there has been evolution of the microscopes to the present era. So is the field of micrometry as well. Sir William Gascoigne, I'm sorry, the slide is moving. I'm extremely sorry for that. Yeah, Sir William Gascoigne is a person who had invented micrometer in early 1600s. And since then, there is also evidence of photography used in micrometry way back in 1876. It was Sir Woodward who had taken measurements of human erythrocytes from 8 criminal individuals, photographed these RBCs, and the sizes of the erythrocytes have been documented way back in 1876. So what is micrometry? So as we understand micrometry, what is it? It is about measurement of cells and its subcellular components, which is derived from Greek word micron, referring to small, meter referring to measurement. It is also known as morphometrics. So morphometrics or micrometry refers to measurement of the size of cells, its subcellular components, or microorganisms using microscope. In a light microscope, the size of the object measured would be varying between 0.2 microns to that of 25 microns. Now, why so? If the size of the object is less than 0.2 microns, it is too less to be resolved under light microscopy. If it is more than 25 microns, we require wide field eyepiece, as we have to use certain specialized type of microscopes like stereo microscopes to measure the size of the objects under microscope. So the measurements thus obtained from the tissues can be expressed in either millimeter or micron. Most common measurement is microns in size for the light microscopic observed images. But for electron microscope, the size is usually expressed in nanometer. The commonest parameters which are measured of the tissues would be length, thickness. We can also measure areas and angles for certain tissues. The commonest methods would be starting with the most elementary method, as we are familiar with our own compound microscope, which is an inbuilt calibrated mechanical stage with an X and Y axis. When a slide is placed on the mechanical stage, the vernier calipers can record the measurement to the nearest accuracy of 0.1 millimeter. But the commonest method, I'm sorry, few slides are moving. I'm sorry for that. So the most commonest method of manually measuring the tissues would be the combined use of ocular micrometer and stage micrometer. Ocular micrometer refers to a simple glass disc which has graduations in the center of it. Usually there'll be 100 graduations etched on it, and this is usually referred as reticle. The graduations on the ocular micrometer is referred to as reticle. When this ocular micrometer is fitted to the eyepiece of a microscope, we can get the readings of the tissue. However, we need to always calibrate this with stage micrometer. So what is the stage micrometer? Stage micrometer is again a glass slide which has measurings etched on it. For a light microscope, the measuring is usually up to each measurement refers to 0.01 millimeter, that is 10 microns for low power microscopy. Why this calibration? Why should we calibrate? That is because when the ocular micrometer is fitted to the eyepiece, when we visualize when we have to visualize the size of the object, it would be varying with the objective that we use, whether we're using 10x objective, 45x objective, 100x objective, or even 60x objective. So there is a need for overlapping of the reticle with the stage micrometer scale to get the actual size or recording of the tissue which we are going to measure. This is what I meant to say, the overlapping of the reticle, wherein we are seeing that the ocular micrometer readings are overlapped with that of stage micrometer to measure or record the size of the tissue manually. Calculations will be done in the form of mathematical formula, where one division of ocular micrometer is equivalent to number of divisions of stage micrometer, which is referred as A, by number of divisions of ocular micrometer, that is B, into 100. So this would say, if we are measuring the length of a particular object, this would be expressed as length in microns. So this is a calculation which is done for 10x. Similarly, we can use a calculation for a 40x or even 45x or 100x objective as well. Where, if we measure the breadth in a similar method, the total dimension of the given specimen in micrometry, manual micrometry, would be length into breadth, which is expressed in microns. This is the manual method of measuring the tissues under microscope. Now, something which we have to keep it in mind is that the recording or measuring of tissues under microscopy is referred to as quantitative morphology, which has two different entities. Morphometry, which we had seen till now, a simpler procedure for measuring the structures with a ruler. If it is a larger tissue, a larger object, we use a vernier calipers or a slide calipers. But when we are measuring the tissues under a microscope, we usually use either ocular micrometer or stage micrometer and record the readings. But the recording which is obtained like this would always be a linear measurement which would include either length or breadth or area. However, many a times we miss out certain measurements of a particular cell component or a tissue component. Like, for example, can we measure the height with the microscope, with the image? Absolutely not, because it is just a section. It's a single section where we are recording. We cannot consider the height. Similarly, we cannot measure the volume of a particular tissue. Here comes the role of stereology. So what is the stereology? It is a method of estimating quantities of a given tissue in the three-dimensional space, analyzing the two-dimensional cut section. Now, this is basically run on a specialized mathematical formula. Now, one more thing which definitely happens during recording of tissue elements is that, suppose the slice is at this level 2, the section is at level 2. Many a times the reading of 1 and 3 and 4 are missed out. In case if a single serial section is not taken into account also, there is a chance of misinterpretation of the actual size of the actual diameter of a particular cell. One more thing which can happen is, I'm sorry for this. Now, let us take one more example here. This is the image of rat pineal gland. The pineal sites here on the left-hand side have been measured using manual micrometry method. Here we are able to see that the maximum diameter D1 is considered as a length, and the next diameter is a D2 as a breadth, and length into breadth gives us the dimension of a pineal site. Now, however, in the same image, we are seeing that we are not able to get the accurate measurement of one more pineal site. It could be various reasons. The tissue section might not be uneven in during the process, during the microtomy. The section might not be taken properly, or during when the paraffin embedded tissue is put into water bath, when the sections are put into water bath, there might be variations in expansion of the tissue. So this results in inaccuracy of readings from the pineal site. For this, we do take into account stereology, which is one more technique where the dissector moves only in one of the planes, either upward plane or downward plane. Now, only those pineal sites which are visible in that particular plane are accountable for measurements, and applying specific mathematical formulas, which are usually design-based formulas, we would take the measurement. So that is about stereology. So stereology is totally a different kind of measurement which takes into account all this. So one more thing which can happen during microtomy is tissue shrinkage. We know that when we take the sections, there are so many factors which play a role. Whether the knife is sharpened properly or not, whether the paraffin wax which is used is able to hold or bind the tissues in a proper way. Is the clearing happening properly? So there are a lot of issues with whether the fixation has happened in a correct way. So tissue shrinkage could happen during the routine staining procedures, and we would tend to miss out the accurate readings from this procedure. And one more thing which can happen is geometric bias. What is geometric bias? The spatial orientation of the cell, not just in reference to length or breadth, it could also be in reference to height, volume, and other features. So there is a geometric bias which can happen in microtomy, and we are familiar that stereology could definitely handle all these drawbacks from micrometry. However, to perform stereology, there are certain limitations. First of all, there is a need of lot of experimental planning. It cannot happen on day one, and we cannot do retrospective analysis from stereology. And there is always a need of skilled stereologists who would help us to do these measurements. And if we are also doing, we should have accurate knowledge about how to go about stereology. Now, the present session is all about image analysis, and micrometry is the basis for doing image analysis from the digital images. So let us not go ahead with how to do about stereology. We'll have a separate discussion somewhere else in future. At this point of time, the replacement for all these drawbacks could be in form of one solution, that is digital image analysis. One more thing which usually, I'm sorry. So one more thing which usually happens during digital image analysis would be, during manual micrometry, as we are familiar with, is visual fatigue. When we see if we have to see probably some 100 slides in a day, obviously our eyes are strained, and at the end of maybe 50th slide, we tend to miss out on readings. And there is definitely inter-observer variability, as Dr. Suma had already pointed out, and there is a role of subjectivity. And one more most important issue is, particularly in digital pathology, which we come across, is abundant glass slides get stored. And in case if we want to retrieve any information about a particular slide, retrieval of information from those glass slides, if it is not organized in a proper way, is an Herculean task. In addition, there are always cognitive traps. The perception of each individual varies about a particular analysis tool. So to combat all this, so there is a shift of quantifying a tissue, that is through manual methods, what is known as manual micrometry, to that of digital image analysis. So digital image analysis is a solution to the issues which happen during manual micrometry. The combination of digital image analysis and optical microscopy has been a boom in the field of biomedical research, particularly translational research. In fact, there are a lot of recommendations who would say now that biomedical image analysis should be considered as a separate subject, and more value should be given to this particular field of research. Why so? We'll come to it shortly. Now, this is a beautiful image which I always cherish. This is digital imaging. So digital image analysis is totally different from digital imaging. Digital imaging, this is a qualitative imaging. Of course, this is an article which is published in the year 2011, wherein there is a specified imaging technique called as brainbow. Figure A here represents wherein they have used fluorescent proteins to identify the neurons. Each individual is given a different color, ranging with the primary basic colors, that is red, blue, and green images. So beautifully, you're able to see the Purkinje neurons of cerebellum. In figure B, we are able to see the dentate gyrus of hippocampus. Figure C represents the neurons of brainstem. Figure D represents the neurons of cerebral cortex, pyramidal neurons, that is third to fifth layer of cerebral cortex. Figure E here is zone CA1 of hippocampus. Now, this is not tissue analysis. It's a qualitative analysis. One more such example, which is not a tissue analysis, would be image texture analysis. What is this image texture analysis? On the right-hand side is an example for image texture analysis. This is done with a specialized microscope called as second harmonic generation microscope. Now, this is a live tissue of an individual, which is imaged through second harmonic generation microscope without using any stains. There are no stains which are used here. And these sections are compared with the routine stained sections. Here, the stain which is used is saffron and O yellow stain of cartilage and bone. With this, the researchers were able to compare the texture of cartilage and bone with these two methods. Again, image texture analysis is definitely not tissue analysis. One more such example would be use of non-invasive technique called as phase contrast microtomography, X-ray phase contrast microtomography of a live individual. In this type of technique, what they have done is they have taken an X-ray phase contrast tomography of an individual, just like how we take CT, MRI or PET images of the lung tissue. They were able to subject the images for quantification of asbestos bodies. The first image is the actual image of the lung tissue with asbestos particles. Figure B is removal of the lung fields and focusing only on the asbestos bodies. Further, they have applied few certain basic principles of imaging, digital imaging, wherein they have filtered the noise as well as removed they have concentrated on the specific size and dimension of the asbestos. And also in the figure D, they have concentrated on the various shapes of asbestos particles. Again, this is one more way of doing digital imaging. But for us of interest would be histological digital image analysis. So what is this digital image analysis or tissue image analysis or quantitative image analysis? It is the extraction of data from digitized histology sections. There are three types of measurements which we can do with this technique, area-based, cell-based, and measurements of other objects. Just to give you an example here, This is the first one is figure A, which represents the quantification of cardiac fibrosis. Now, the left-hand panel is the routine hematoxylin and eosin stained sections of the heart tissue. Here, the eosin stained is the cardiac myocytes and the hematoxylin stained would be the fibrosis. However, the right-hand panel of this image shows the applicability of digital image analysis, wherein we can exactly record and measure the area of fibrosis, which is represented by blue color here. Yellow represents the unstained. So every detail should be provided to the software to record. That is also a message here. It is not just counting it. There is a role of a researcher who would feed the information to the computer. Similarly, this is figure B, which represents differentiation of two different cells. This is the section of uterus. Here, glandular epithelium has to be differentiated from stromal cells. How is it done? It is done by focusing on the nucleus, particularly the alignment or axis or orientation of the nucleus helps us to pick whether it is the glandular epithelium or the stromal cells. So one such example of how digital image, and if we have to manually count, either we miss out or we might even take a lot of time to do this counting method. But however, digital image analysis can do in fractions of few seconds and give us the findings. Now, this is the one which is a boom in the field of digital image analysis. What is that? Particularly identifying certain special cells. Now, the left-hand panel of this image is immunofluorescent stained tissue, which has identified the tumor cells. Now, the dark brown colored cells are the tumor cells. However, many tumor cells are missed in this particular image, which are immunohistochemistry negative tumor cells. There comes the role of digital imaging. Now, the right-hand panel would be an example of the applicability of digital image analysis, wherein the brown-colored nuclei. So here, basically, they're focusing on the nuclei. The brown-colored nuclei represent the immunohistochemistry positive tumor cells. There are many blue-colored nuclei here, which represent the immunohistochemistry negative tumor cells. The green nuclei represent the stromal cells. So we can quantify, we can count the number of all the tumor cells, and usually it doesn't get missed out if we use when we apply the digital image analysis. So what are the other applications? Is it just that? No. There are a lot of researches which have been done so far in the field of digital image analysis. Just to give you a few illustrations from the literature, they have quantified the lung metastasis. In fact, the digital image analysis has been a boom in quantifying the angiogenesis, that is counting the number of new blood vessels and lymph vessels in developing mammary gland, quantifying intratumoral heterogeneity, quantifying liver fibrosis and hepatic steatosis, quantifying immune filtrates, particularly the lymphocyte subpopulation can be quantified. And in the field of genetic, it has made a remarkable progress where it would help us to quantify the mitotic figures. In the field of digital pathology, of course, to quantify biomarker positive cells, Ki67 positive cells, which would definitely tell us and help us to differentiate or help us to stage, especially when there is a dilemma between stage 1 and stage 2. This digital imaging would definitely be an additional method for us to come to conclusion. So some more importances of digital image analysis. So why should we quantify the images? Some more applications, as in recording or measuring the cellular morphology. For example, if we have to screen certain cells for drugs, we need to quantify the morphology. We need to record the shape, size, dimensions of the cells, and what is their effect on the drugs. So what is the effect of drugs on these cells? Then comes the role of digital image analysis. Second important use is when we have to classify the cells in an image. For example, when we have to see specifically whether a particular stem cell is differentiated or not, we can use image analysis. When we have to do image specification, we have seen a lot of examples so far. If we have to differentiate immunohistochemistry positive and negative cells, we have to differentiate a glandular epithelium from stromal cells. Similarly, if we have to differentiate epidermis and dermis, so specifying a particular tissue element is very easy with digital image analysis. Or digital image analysis. One more application of the digital image analysis is in object tracking. For example, when a live cell has to be tracked in an embryo, now this image analysis comes to play. What are the strengths of digital image analysis? One is, for this, automation and precision. Automation refers to increasing the capacity. Now, within a span of few minutes or few seconds, we can get the results or we can get the measurements of, say, some 100 or even n number of slides, or the whole slide imaging can give us the results. And one more thing is precision. One more advantage of digital image analysis is precision. Every time an image is subjected to analysis, we would get the same readings. That is not the case in manual measurements. Our own eyes might give different interpretations at different point of time or at different, it could be due to various reasons. And it could be even inter-observer variability also. That is not the case with digital image analysis. Precision is the requirement. However, precision should be differentiated from accuracy. So what is accuracy? The values should be as near as possible to standard values. There comes the role of a researcher. The researcher has to determine whether the image analysis which is obtained from the software is accurate or not. It is precise, agreed, but whether it is accurate or not requires the role of a researcher. What are the commonest tissues which can be subjected to digital image analysis? I'm sorry. Again, so it could be all possible tissues, like for example, formalin fixed and paraffin embedded tissues, cryosections, whole embryo sections, organ wounds. All possible tissues can be subjected to, all possible tissue preparations can be subjected to image analysis. Similarly, all possible stain sections can be subjected. Routine hematoxylin and eosin stain sections, histochemical stains, chromogenic immunohistochemistry stains, fluorescent labeling stains, in situ hybridization stains. In fact, we do have softwares which would ask us which stain are you using for analysis. So we would start with that particular step for analysis and processing. The basic steps of digital image analysis would be image acquisition, then followed by image processing, then image analysis. Image acquisition, as it is as simple as that. Whatever tissue which is focused under a microscope is acquired. The image of this would be acquired with specified cameras. In addition, if we are interested in research and if we are interested in analysis, we do have a database which is available in either public domain or private domain, where we can do the necessary analysis or quantification with this image acquisition. Now, this image acquisition step also requires some amount of pre-processing. What the images which we obtain might vary with color or with illumination. So normalization of this is the need of hour. Then image processing. The image processing step includes certain steps wherein it would be helping the researcher to do the further step of analysis. Like for example, feature extraction, segmentation, and as Dr. Suma had already pointed out, image enhancement. So all these are the image processing steps. Once the image processing is done with specific software tool, the next thing which is done is image analysis. Image analysis would be of 3 different categories. We can do low profile image analysis, medium or moderate profile image analysis, or a very high profile image analysis. Low profile image analysis is simple measurements of by linear methods, like measuring the length, the breadth, area, the thickness of a particular tissue, cellular element. It would be a low profile. Medium profile image analysis is classification of the tissue elements. We have seen a lot of examples, whether it is immunohistochemistry positive or negative tumor cells, etc. But high profile image analysis is the diagnostic method. Now, without manual observation, if the software is able to diagnose what type of tumor it is, what is the diagnosis, what is the stage of that particular tumor, that would become a high profile image analysis. And here comes the role of the latest boom in the field, that is artificial intelligence, particularly a branch of artificial intelligence or a subtype of artificial, that is deep learning. We'll see that shortly in the subsequent slides. So let me recapitulate here. The 3 basic steps of image analysis, digital image analysis, would be image acquisition, image processing, image analysis. And most of the time, if we have a limited or a resource poor setting, we can do both low profile and medium profile analysis. However, for high profile analysis, definitely we require a very good software support and background to do high profile image analysis. Now, as we move on to the digital image analysis details, there are a lot of terms which we need to understand. Now, when we did image analysis on particular tissue, neuronal tissue, it took us a lot of time to understand these terms, to be familiar with these terms. So I thought, let us first see what are these terms and what are the basic things which we have to keep in mind when we are doing digital image analysis. To start with, binary image. Binary image. What is a binary image? Whatever image, this is a slide of liver tissue, normal liver tissue. Now, whenever we subject this image or process this image to the software tool, the image analysis tool, the images are categorized into 3 basic colors: red, green, and blue. And each recording of this is given in what is called as binary bit or 8-bit, or bit. Binary bit refers to the value which is given for the pixels in the image. Now, in other words, it is converted into a black and white image in a simplest term, but it is not so as simple as that. The values for this image is given in the range of 0 to 1. 0 refers to totally black and 1 refers to totally white image. Now, when we use all the 3 colors are illuminated simultaneously. That is, red, green, and blue are illuminated simultaneously. It gives us a very black background, black image, that is 0. If all the 3 elements are switched off in the software, we get a value of 1, which is a white background, white image. So a combination of 0 to 1 values would be expressed in a binary image. Why this binary? There are a lot of advantages of it. First of all, let's see with this slide. Definitely, we can count better if we can quantify an image better if we have this kind of image compared to a routine hematoxylin and eosine stain section. This is of a neuronal tissue. So the arborization of the dendrites and axons come in our way. And it is if we have to record the measurements of the nucleus, it becomes it is difficult. So that's one of the advantages of a binary image, where the exact measurements would be obtained. And particularly when we apply a grid, it also helps us to count. So one of the processing steps is applying the binary with grid to record the measurements. And to add to this would be outlining the image and skeletonization. This is not a necessary step for all the images. This is one of the steps which can be particularly helpful for neuronal quantification. When we have to quantify the neurons, what they do is they take out one layer of pixel. One layer of pixels, when it is taken out, the total, some more amount of clarity is brought into the image, and the thick lines become much more thinner. So by skeletonization of the image, we can do a lot of analysis pertaining to neural tissue, like shoal analysis, fractal analysis. These are all recordings which can be done with this image software, imaging tools for the neuronal tissue. That's about one more term. So there are many more terms. I would like to use a video here to show you basics of image analysis. Hope the video is visible. Is the video visible?
Yes, ma'am, the video is visible.
How about the audio part of it?
Just play it, ma'am.
Now?
Audio. It's not coming, ma'am.
It is not, is it? Just a moment.
No,
just a moment.
Audio is not coming, ma'am.
No?
No, ma'am.
Just a moment.
Yes.
Now, I'm going to play it now.
Tricks that people who are coming into the imaging facility, either as
Yes, ma'am.
new honors
The audio
students
is
or PhD students, or people who've done microscopy before and just wanted a refresher, to have quantification, and so knowing what to do is really important. Image analysis does not equal image beautification. So when we do things in Fiji or any of the commercial programs that we have, we're not just trying to make the image look pretty. It's not Photoshop. You shouldn't be using Photoshop for any of your microscopy image processing or analysis. An image is just an array of numbers at its simplicity that represent the intensity that was detected by the digital capture device. So in this example of a nuclei, if we keep zooming in, we'll be able to see that there's just discrete pixels that have gray values that correlate to these numbers. And a pixel is just a picture element. So that's the 2D representation. But in when we're doing 3D visualization, especially, and when we capture data on the microscopes, there is volume information with each slice that we take. And that we refer to the 3D pixel as a voxel or volume element. And in a classic fluorescence microscope image, if we look at it in like its numbers form in a spreadsheet, you can see that all these background values are quite low, and that's the black values. And the object in the foreground, the nuclei, will have these higher values. And there's two main types of images that we will take in the on the microscopes. There's RGB color images, and these are good for capturing the histology and bright field images, and we capture them with color cameras. Or we capture in grayscale or monochrome for our fluorescence. Now we can have multiple fluorescence images that we all take as 3 or 4 or however many channels of monochrome, and then combine them to see multiple colors. But this isn't an RGB color image, it's different. So when we talk about capturing our grayscale or monochromatic images, they have a bit depth. And this is like a color resolution, or the gray levels of the image. Most of our systems will by default capture in at least 8-bit. There are higher methods, so 12 and 16-bit, and these are better for quantification. And one important thing to note about gray values or bit depths is that they have to be absolute. There can't be negative values. So if you have a 1-bit image, it's 2 to the power of 1, and you have 2 options, black or white. For 2-bit, you would have 4 options, and 3-bit, 8 options, right through to the ones that we're more realistically capturing, which isn't in these discrete bins. It's 256 levels of gray, 4096 for 12-bit, or 65,536 for 16-bit images. When we take our monochrome images, we don't have to display them in this grayscale. We can have different lookup tables, and these are just how we map the color onto those intensity values that we've recorded. So if you have a dark background and light objects in the foreground, and you're capturing an 8-bit, you would see that this gradient goes from black being at the background through to pure white, which would have a value of 255, or some derivative in between. We can invert the lookup table so that we can say that our background is actually 0 and our objects in the foreground are dark. And that can be useful for displaying really fine structures, because we can see dark things on a light background a little bit better than light things on a dark background. And we can map colors to them. So if we have a green channel image and we want to show like our GFP as intensity of green, pure green would be mapped to 255, back down to 0 for the background or the dark bits. Now, when we apply these lookup tables, it's important to think about color blindness and how it's displayed can be, may not be what you see, what other people see. So if you have a red sensitivity for color blindness, where you'd have your classic red, green, and blue image, then people might find that hard to see because it would appear like this. Whereas if we modify the red channel and display it as magenta, then this is how it would appear to people who are colorblind. And we can still see these differences where they're lost in this image. And one of the main programs that I use, Fiji, has a really nice tool to simulate color blindness, and you can have a look at.
So I would like to stop this video at this level because the rest of the video is more about the camera settings and the values. I would definitely be sharing the details of this. It's a
all the different ones.
It's
and you can
available
see
in
how the images will appear.
The YouTube. So we will stop here. Just a moment. To continue, so we understood certain basic components of how the image analysis is done, or basic terms related to image analysis. So what is this image analysis all about? It's about use of specific software tools. So to name few tools which are commonly used in digital image analysis. Just a moment. Yeah. These are the 7 most useful tools which are available online. These are all free software tools. That means to say they are open source platforms wherein we can download these softwares and readily do the analysis. In fact, few of these software tools also have ready-made images available for us, available with us, so that we can take the recordings and proceed further. So to name them, the commonest one, the workshop is which about all, today's workshop is all about ImageJ Fiji, QPath, Cell Profiler, Elastic, Orbit, IC, and Cytomine. ImageJ, we had an introduction on this Java-based image analysis program, which was developed at National Institute of Health in collaboration with the Laboratory of Computational Instrumentation at the University of Wisconsin. Now, whenever an user uses ImageJ software, validation and verification has to be done from the end user. Fiji is an plugin specifically for with the ready-to-use plugins available. So always we use a combination of ImageJ Fiji for our tissue image analysis. The latest one is ImageJ2. We can crop, we can do all pre-processing techniques, we can quantify the images, we can classify the fluorescent imaging, not just fluorescent, all kinds of, starting from hematoxyl and eosin stain sections to that of in situ hybridization stain sections. We can do the analysis with this particular software. Just to have an example here, this is the semi-quantitative technique. That means to say there's some amount of manual intervention which is available here. Now the cleared area in the center, this is the lung tissue of a mouse, where they have measured the area of, they have measured the area of the alveoli. Now, particularly the clearing in the center is done because of the vascular tissue. They don't want the alveoli which are nearer to the vascular tissue. That is one of the steps in their research work. So it is the ImageJ software helps us to analyze this area of the alveoli of the lungs in this particular study. Cell Profiler. So I'm not going to go to the depth of image analysis, how it is, because the subsequent sessions would be in detail about ImageJ. Cell Profiler or along with Cell Analyst. Cell Profiler is the second most commonest image analysis tool available in the public platform. It was developed by Broad Institute of MIT and Harvard It's a MATLAB-based free software which enables scientists to analyze batch of slides, that is batch process of cells and biological images. Cell Profiler runs on modules. So there are a lot of modules for loading the image, applying the identifying the primary objects. If we have to identify nucleus as primary object, cytoplasm becomes secondary object. So identification of the primary objects, then subjecting them to quantity or analyzing them. Or the collection of such modules become a pipeline. So Cell Profiler runs on a concept of pipeline. There are a lot of pipelines which are available, ready to use pipelines, and whichever component of pipeline we need, whichever module of pipeline we need, we can use it for our reference. For example, when enhancement is already done for a particular image, we can skip that step, or we can delete that particular module. So there is a provision for either adding or deleting the module in Cell Profiler. Now, this is Cell Profiler Analyst. I just like to mention that when we started doing image analysis with Cell Profiler software, we couldn't analyze the images, we couldn't process further because we had downloaded this Cell Profiler Analyst. We didn't know the basic difference between what is Cell Profiler and Cell Profiler Analyst. Cell Profiler Analyst is only data analysis software. Whatever analysis results that we get from Cell Profiler software is further processed and executed from the data analysis software, that is Cell Profiler Analyst. Now, this particular software is extremely useful when thousands of slides are analyzed simultaneously, and we need to get the values of large quantity of images. The data is processed and classified and grouped, and the values are given from this data analyst software. This is a classical example of a pipeline. This we had applied to our neuronal tissue. On the left-hand panel, we have input modules, which includes images, metadata, names, and types and groups. Once we want all these input modules, we also would be putting the analysis modules, whether we will have to enhance or suppress the features, whether we need morph. Morph refers to the shape of the neuron, identifying nucleus as a primary object. Then once we subject, now here the red signal represents that this particular pipeline cannot do the overlay outline module. So it cannot run this particular step. So whatever pipelines recordings that we require, once we subject it to analysis, we would get the necessary results, and the results would be like this. This was a pipeline which was for recording the values of a primary object, where the nucleus was considered to be the primary object. And we had got the recording of the primary object, that is nucleus, at 10th percentile diameter, median diameter, 90th percentile diameter. And all these values were in pixels, and area covered by objects, that is 2% of the total area covered by objects. That also we would get from this. So one of the examples of how a Cell Profiler software runs. Now, this is particularly useful for biological tissues. Unfortunately, ImageJ and Cell Profiler cannot go hand in hand, but Cell Profiler has lot of integration tools. Now, the rest of the image analysis tools, which I was mentioning earlier, come into this category of integration tools. IC, Elastic, Orbit, Cytomine. These are some integration tools. What is exactly meaning integration tool? To start with IC. Now, this is actually a Photoshop software, but it has upgraded itself to image analysis software, where it can process and pass on the information to Cell Profiler, and we can do the measurements. Elastic is one more collaboration between Cell Profiler and Cell Profiler with Elastic. Now, this Elastic software has two advantages. It is very user-friendly. Someone like me, who doesn't know much about software, could definitely use Elastic as an image analysis tool much more easier. And whatever information which is obtained in Elastic, there is an element of feedback here given from the Elastic. So it would tell us which step are we not doing correctly. Are we doing the enhancement correctly? Are we doing the segmentation properly? So that kind of feedback is given from Elastic. So, however, particularly in the field of the latest field, that is deep learning, they are doing a lot of research. The Cell Profiler has integrated with Elastic to bring about the applicability of deep learning in image diagnosis, well, as a diagnostic tool. Orbit is again an integration tool with both Cell Profiler, and in fact it works with even ImageJ. Again, huge batches of slides can be analyzed. It runs on a concept of tile-based processing. Cytomine is particularly a tool which would help us to quantify the images when we have the whole slide. So it's one of the whole slide imaging tool, and these tools help us to stitch or stack the images together and bring about a meaningful information. This software tool, QPath, is the favorite tool of a diagnostic pathologist, digital pathologist. It was invented by Pete Bankhead, and it's a whole slide imaging technique, particularly useful for quantification of biomarker positive cells, biomarkers, immunohistochemistry quantification of a tissue. It has an inbuilt whole slide image viewer, which automatically detects the cells and classifies the cells into subtypes. The data which is obtained from QPath can be exchanged with ImageJ, and the combination of it can be used. Here is an example of how QPath works. Now this is the normal kidney, of course, which I had the image had been downloaded from me to show you, but it is not of a tumor tissue. As we can see here, left hand side, it is giving us that there is none of these elements which are present in this normal slide. What are those elements? There's no tumor tissue. There is no stroma pertaining to tumor tissue, of course. Immune cells. Then the immunohistochemistry positive and negative cells are absent. So if we have all these features in a particular slide, we can analyze it and give in meaningful information about the tumors grading and staging also. As I had earlier mentioned, we can do digital image analysis for all possible stains. Here's one example. When we have to do image analysis, it would specifically ask us which kind of image are you detecting, whether it is hematoxylin and eosin stained image. Are you doing image immunofluorescent imaging? If we are doing if we are subjecting the image taken from immunofluorescent stained slides, it would do a different kind of processing. For example, for green immunofluorescence, there is a separate processing of enhancement. For blue, it's a different kind of enhancement, or a combination of it is it has a different kind of processing steps. So that is an advantage of doing using this QPath as an image analysis tool. So bioimage analysis has revolutionized the field of quantification of the neurons also. So I would like to touch upon this deep learning technique a bit later. Let me finish off with two more important slides pertaining to image analysis, particularly the neuronal tissue. Neuron Studio is the most commonest freely available tool which can automatically trace the neurons. We can trace the dendritic arborization, we can count the spines, dendritic spines, we can get meaningful meaningful information about the morphology of the dendritic tree of a neuron. We can subject the image to shoal analysis and count the spines. Now, what is this shoal analysis all about? Now, unlike the other tissues of the body where there is a definitive shape and size for a cell, neuron is not so. And recording the measurements of neuron is totally different from recording the measurement of any other cell. Just like how the ball ball is definitely circular, and it is easy to record the diameter of a ball. But when it comes to a tree, how do we measure the dimension of a tree? There comes the role of shoal analysis and fractal analysis, and even ImageJ can do these particular analysis. But Neuron Studio has advanced features of doing quantification of neuronal data. L-Measure is one more such quantitative morphological tool, image analysis tool, which would give us all the possible informations about the neurons, be it width, height, depth, diameter, section area, Euclidean distance, branching order. In fact, we can even record the angle which is between two dendritic branches. So that is the applicability of this particular image analysis tool, which is known as L-Neuron. So this is the summary of various image analysis tools which are available for us to use and go ahead with tissue quantification. Now, these are the most important hubs or scientific hubs, particularly the first and the second one, which I had used a lot for understanding what is image analysis, how do we download the various softwares, and how do we do the various steps. In fact, we have PowerPoint presentations, we have manuals, we have videos to support, and all possible support is available from these resources to carry out the quantification of the images. A word of caution: whenever we are using image analysis, please keep it in mind that it is not the software which is making the measurement, but we are doing. That is because the kind of information which we are processing or giving to the software will help us to get the meaningful result. Okay, precise calibration is the need of our. Now, what is the meaning of precise calibration? Even the calibration should be done for all possible objectives, whether it is low power, high power, or electron microscopic, high power or oil immersion objective. So for all possible kind of objectives, calibration is the need of our, otherwise we will not get the desired values. The image size and format definitely affect the measurements. Image size, whether that is usually expressed in pixels, pixel per element, and format, what we meant to be a format, as Dr. Suma had already mentioned, it could be JPEG format, PNG format, TIFF format. Ideally, most of the softwares prefer TIFF format, not the JPEG format, which we commonly use. JPEG will give always, with each analysis, there is losing of data. So none of the softwares prefer JPEG, but they usually prefer TIFF format for recording the measurements. We are familiar that tissue undergoes a lot of dimensional changes during tissue processing, during paraffin embedding. So whenever we do analysis, these things, there is a need of adding a correction factor for the factors for especially paraffin embedded tissues. So ideal way of doing measurements is using epoxy resin embedded tissues, which is said to be giving us a less amount of dimensional changes compared to paraffin embedded tissues. And of course, we need to use appropriate reference points whenever the measurement is done. So to summarize, digital image analysis is an ancillary method of micrometry. Ancillary method, and of course, it can definitely be a substitute for manual micrometry to a great extent. Just like how we have inter-observer variability with manual micrometry, we do have inter-algorithm variability, which always has to be considered while doing meaningful measurements. The Achilles heel of digital image analysis is objectivity or accuracy. There is precision with digital image analysis, but objectivity of the values is determined by the researcher and not by the software. So there is a need for synergy of an experienced researcher and precision and consistency of the optimized algorithm. That would definitely lead to robust and reproducible results. Deep learning tool application has a substantial promise. I'm sorry, due to paucity of time, I couldn't play a video where how exactly the deep learning has evolved, especially one of the steps where image segmentation is being done. If the host has additional time, I can share that video after this session. Early engagement of researcher is the need of our. So not that we come and just do the analysis, run the program and do the analysis at every step, be it image acquisition step or be it pre-processing step. We have to calibrate, we have to use the correction factors so that we would get the accurate results from digital image analysis. So with this, I would like to end my talk. I would like to express my sincere thanks to the organizing committee for giving me this opportunity to share few of my views. I thank all the delegates for your patient listening. I would like to express my thank to my institution for their support and constant encouragement in giving this lecture. Thanks to one and all again. Thank you.
Okay, I think I should thank Dr. Vasudha for a very informative and exhaustive discourse, I should say, over the micrometry, starting from the very basics of what micrometry is and the fallacies of making measurements on thin sections. And she has touched upon every aspect of the digital image capture, digital image segmentation, that is feature extraction, and the digital image analysis and the measurements, and various, she has also touched upon various software, more importantly, from the Free Software Foundation, that is the free and open source software. And I think for many of the audience, this must have been very overwhelming. Some of us who have been working in this field for decades could follow most of the things that Dr. Vasudha has discussed. And I think I just want to emphasize that anatomy is a visual subject, and digital image is the very basic thing that every anatomist should understand, because microscope is the tool of the anatomist and the images. And of course, there are other sources of images which anatomists use, from the CT scans and MRIs, and basically digital image from any source is same. And she has also explained in terms of pixels, that is a picture element. And if they are 3D images, those are actually stacks of 2D images. And in that case, you refer to the same pixel of every slice of that particular stack as voxel. That's the only difference. And she has also touched upon the very basic way how a pixel is represented in a digital image. A pixel is actually the smallest element, or the dot in the image, which can have a value. It can have a value of 1 bit, where it is either black or white, that is 2 values, 0 or 1. It can have more number of bits. If you use more number of bits, more values will be possible between the black and the white, and that is called a grayscale image. So there is a binary image where the element is either black or white, and there is a grayscale image where the picture element can have a value from 0, that is black, to 255, that is white. That means a total of 256 values. And the color images that you see are actually 3 grayscale images. And the gray value in each of the channel is representing the red, green, or the blue. And when you combine these, you get millions of colors. So the color images are, every panel, that is red panel, green panel, and the blue panel, has a value of, which is represented by 8 bits, where 256 values can be represented in 8 bits. So now I think that is the very basis of a digital image. And then once you have this, you can do anything with the digital image, because for a computer, the image is not a picture. It is just a table of numbers. And for you and me, it appears like an image. It just shows a collection, an array of pixels. And the video that Dr. Vasudha has pointed to is very informative, and all of you must see that, and everybody should have a handle on what exactly is a digital image. I think so. I congratulate Dr. Vasudha once again that this is a beautiful talk, and probably this is the first time that I have seen. I hope you are also an anatomist.
Yes, sir. Yes, sir.
Yes, sir. Yeah. First time I have heard an anatomist talking so much about digital imaging, image analysis, and micrometry. And
Thank you so much.
Yeah, and you touched upon stereology. I think more than micrometry, it is stereology. There are so many tools of stereology available in the image itself. There are plugins by which you can do stereological measurements. And I also want to point you to a very good book on stereology by a Turkish anatomist, and whose name is Kaplan. I think many of you, So I recommend this book to all of you, and please read and understand the principles of stereology. And they are very simple. They are mathematical. They are actually statistical methodologies of estimating the volumes and the areas and the dimensions of things which exist in three-dimensional space using two-dimensional samples, that is our tissue sections. Okay, so if you do just simple direct measurements of length, breadth, diameter, circumference on single slides or single sections, they can be fallacious, as Dr. Vasudha has pointed out already. Supposing you have slices of a ball, and you will get circles of different diameters, starting from 0 diameters if the section has gone tangentially to the ball, and if it has gone through the middle of the ball, the largest diameter. That's what she has illustrated. I hope everybody understood that. The fallacy of measuring diameters in the sections is very false, because if I give you all the slices of a ball and ask you to make measurements, and then most of the people generally take an average of that, and that will always be lower than the actual diameter, because there are more sections which are representing the smaller diameters of the ball than the largest diameter. Okay, so anyway, I think that is where this stereology comes in. And I am very happy that Dr. Vasudha has pointed out the importance of all these things. And from now on, every anatomist will keep in mind how to make measurements which will actually represent the truth. Okay, yeah, good, very good. And I congratulate, thank you. And I'm sure there are a lot of questions in the minds of the audience, because this is a very extensive, very exhaustive topic. And actually, each of them deserves a separate CME on its own. For example, digital imaging, image capturing and image segmentation and all those things can be a separate CME, and stereology can be a separate CME. And then coming back, she also touched upon the artificial intelligence, deep neural networks. Actually, these right now, they are all black boxes. You give the input of one image from one side, and the other side, it says, okay, identify all particular cells on this side. It will identify and tell you. And most of the time, you won't even know what criteria it is using. So it is a black box. So artificial intelligence has to be used carefully, because if there is a slight difference in something, then there may be totally fallacious results. Using, I think there was a very famous example of artificial intelligence identifying the enemy tanks in a battle. And I'm sure many of you have seen. And just when the time of the day or the season changes, everything has gone haywire, because it was not exactly using the image of the tank to identify the tanks. It was using everything else. Okay. So anyway, I think these are the cautionary words. But the thing is, this is the right time for all the anatomists to rejoice, because there is digital imaging and there are so many tools we can use for analyzing and measuring, because science is nothing but measurement. If you can measure accurately, you are a good scientist.
Thank you, sir.
Okay, yeah, thank you. Thank you so much, Vasudha. I congratulate you once again.
Thank you,
yeah,
sir.
and I also thank the organizers for giving me this opportunity to chair this session. I am really proud of chairing a session which is being conducted in my own very alma mater. I'm still proud of my own alma mater. Okay, I even now I keep telling that JIPMER is the best undergraduate or postgraduate medical school. Sitting in the apex institute of the country right now, I still hold that respect and regard for my own alma mater. I am proud of it. Thank you.
Thank you very much, sir. Thank you very much for your valuable comments and suggestions, sir. It feels a lot of like for hearing this from you. I call upon Dr. Daisy ma'am to give her valuable suggestions and comments.
First of all, I want to congratulate Dr. Vasudha for giving such a lucidative, such a information and a beautiful presentation, beautiful presentation.
Thank you, ma'am.
And really, and it was a need of the time. And as Dr. Sharif has touched upon many points which I wanted to touch, but I won't repeat them. But he has given a very nice commentary on your lecture. It was beautiful. And you have given started with the history, evolution of micrometry, and then you gave the micro details of the micrometry. Then you compared the manual and digital, and then going further and further. I really appreciate. I really appreciate. And then you gave the applications and uses of digital image analysis by giving beautiful examples and nice, beautiful slides. You have really worked very hard. I can understand you must have taken many, many days to prepare such a lovely lecture, really. And then you touched upon the latest methods, that is neurostation and Scholz analyst and QPath and new tools. And it is very good information for particularly for the younger generation and those who want to do research in histology. And it's a beautiful information you have provided. And I agree with Dr. Sharif that such a lecture I've never heard before. And really beautiful, beautiful. And I want to congratulate the team and Dr. Soma and Dr. Rajesh, and all the organizers for organizing such a lovely, beautiful, informative conference, CME.
Thank you.
Thank you so much. I'm
Thank
so
you, ma'am.
glad that
Thank you very
they
much.
asked
Thank
me
you, ma'am.
to be part of this CME. Thank you so much.
Thank you very much, ma'am. Thank you very much. So, due to the paucity of time, I would like to take only 2 questions from the audience, and I would like to present to the speaker. The first question is, what if there is overlapping of nuclei? Another question also same, cells and nuclei if there is overlapping during quantification. This is first question, ma'am.
Okay. Actually, I wanted to show this overlapping of cells and nuclei through a video of how we do a procedure called as image segmentation. Now, whenever there is, let me just put it in words. If usually how the software does is, they compare all the image, all the cells to a place. So image is like a land or a plateau where each cell is considered to be like a mountain. So they consider the peak of the mountain. From there, if the water falls down, they would span out and it occupies a particular area. So using this principle, what they do is they segregate first the cells, and from that, the nuclei are picked up. Or if there are only nuclei, the same principle are applied. When the nuclei is highlighted, the same principle is applied. I would like to share in the chat box the video which I wanted to show about image segmentation and especially the application of deep learning in this. So it will be helpful for the participants to know the details of it. Yes.
Thank you. Next question is it possible to do whole slide analysis in image J?
Definitely. Image J, at this point, I have not gone to that level. My understanding is that whole slide imaging is better with cell profiler than image J. I think either Dr. Yogesh sir or Sharif sir could answer this question. Can we do the whole slide imaging? Yes, sir.
Yeah. See, for the actually whole slide imaging is one thing and analyzing that image is another thing. Okay,
Yes,
whole
sir.
slide imaging, you will have to capture the image in multiple fields, and that all those fields have to be stitched together to get a single image. So it is possible to do this particular stitching using many tools. There are tools which are actually available even for free. I think Panorama Stitcher and all those kind of things. Okay, so you should be able to. In fact, most of the cell phones are able to do that now in the sphere photography and all these 360-degree photography. What they are doing is they are taking all those images and they are stitching together. Only thing is there has to be certain amount of overlap on the edges of each of the section. 5 to 15% overlap has to be there for that to be stitched correctly. So whole slide imaging involves capturing of the images in multiple fields, and then each field should overlap with the previous field. Okay, and you have to make sure that each field is overlapping largely, at least 15% overlap has to be there. In that case, and then when you come down to the next row, like you will be scanning horizontally, then you will come down and then go from right to left. again come down and left to right. So this is how, like a grid, no, you have to scan like this, capture all these images and those. Now that is how whole slide imaging can be done manually. But there are tools like Dr. Vasudha has already pointed out, whole slide imaging can be done. The tool will automatically keep capturing. It will show you markers where you should keep the next margin of your field. So if you keep on following those markers, it will capture those images and form the whole slide image. And ImageJ can analyze, you can use ImageJ for analyzing anything. And she has also pointed out that you should not use JPEG. JPEG is a compressed file format, and every time you compress, you keep losing data. So the actual pixel values are getting lost every time you compress. So uncompressed ImageJ formats are available if you want to use that. Otherwise, TIFF is the best format, tagged image file format. It is an uncompressed file storage. Only thing is the files will be a little larger. So it is always good to have the data always, whether you use it or not. Because if you have sufficient data, you can always discard it any time. But you cannot recreate the data which is not there in the image. Okay, I think these are two important things. Using the correct file format is important. Okay, ImageJ can handle any kind of images, even stacks of images from CT scans, MRIs, even if you take serial sections of your this thing, it can handle the whole stack. So if you use the stack and if you know exactly what is your section thickness, you can also calculate the height of the objects.
Yes, sir.
That's what Dr. Vasudha has pointed out, that height is one thing that you cannot get from a single section. But if you have a stack of sections and you know the thickness, you can also get the height of the object.
Yes. Very nice, sir. Very nice.
ImageJ is a very powerful software, and there are lots of plugins available for ImageJ, cell counters and all those kind of things. And Fiji is ImageJ plus all the plugins that are available. So it is recommended even by Dr. Vasudha that Fiji should be used, not ImageJ, because you may not know what to do which particular plugins to use or to include. Because you will have to search for the plugins and download them and then install on your computer.
Yes.
Instead, Fiji will come with all the possible plugins.
Last question: how to combine QPath with ImageJ?
Sir, in fact, QPath has ImageJ plugin, ImageJ collaboration. In fact, can I share my screen? Is it still visible?
Yes, ma'am.
So it would take time, but
no problem, ma'am. No problem.
You can
just
share,
say,
ma'am. You can share.
Okay.
Please share this.
Okay. In that case, I will show you how exactly we can use the. I think no. Okay. If I use my desktop, I think if it is possible to use my desktop, I can show. I have downloaded the QPath.
Okay.
No. No, I'm not able to share my advanced, right? Yeah. Let me tell them, let me tell the participant that QPath, when we subject the images to QPath software, on the right-hand menu bar, there is an image analysis. It would definitely ask us, can we do can we use image analysis for this? So it's collaborated, not an issue at all. Once you start using, you'll get to know that we can use it definitely.
Okay. There are many more questions in the chat box, ma'am. We will compile the questions and we will send it to you.
Sure,
and
sir.
After obtaining the answers from you, we will pass it on to the participants, ma'am. Because this session and elaboration on itself requires nearly more than half an hour, and we had other sessions online. And yes, I sincerely thank Dr. Sharif sir, because ever since my PG days, I used to rejoice the discussion with Dr. Sharif sir on various aspects of anatomy and various aspects of life as well. And sir always gives a totally different perspective to whatever topic we have in our mind. And still, today I got to remember, like the session was on one dimension, means the explanation which were given by Dr. Sharif sir totally uplifted it to another dimension, and the session became like multi-dimensional at the end of this interactive session. Thank you very much, sir. Thank you very much for your wonderful input, sir.
Thank you.
Yes, sir.
Thank you.
So now, yes, sir, I would request Dr. Sharif sir and Daisy madam to unveil the certificate of Dr. Vasudha Kulkarni ma'am for her presentation.
Thank you, sir. Thank you, ma'am, for this great opportunity. Humbled to receive certificate from you both. Thank you so much.
Thank you, ma'am.
Thank you.
Yes, ma'am. Now it's time to unveil the certificate for the chairpersons, and I would request Dr. Suma ma'am to unveil the certificate for Dr. Sharif sir.
Dr. Sharif sir,
Yes, ma'am.
thank you very much for
You're
giving
most
your
welcome.
valuable input and taking the time of your busy schedule and coming online, sir. Thank you very much, sir.
Yes.
You're most welcome. Thank you.
Next,
It's a pleasure.
I would like to unveil the certificate for Daisy ma'am.
Thank you, Daisy ma'am, for spending your valuable time. And I heard that you are like in USA. Thank you for sparing your time for this session, ma'am. Thank you very much, ma'am.
Thank you, ma'am. Thank you, ma'am. So with this, we will be proceeding on to the next session. The next session is a live demonstration, and the topic of the live demonstration is installation of image software and system requirements. It will be rendered by Dr. Gladwin, Associate Dean Academics and Additional Professor, Department of Anatomy, JIPMER. For most of the anatomies, introduction to Gladwin sir is not required because he is one of the very noted person all over the country. And Dr. Gladwin sir is an MBBS, MS Anatomy and DNB Anatomy graduate. Sir is currently additional professor in the Department of Anatomy. In addition to the existing responsibility, he is the Associate Dean Academics, JIPMER, Puducherry. Sir has done his MBBS from Madurai Medical College and post-graduation MS Anatomy from JIPMER, Puducherry. Sir has a profound undergraduate teaching experience of 16 years and postgraduate teaching experience of 11 years. Sir has 20 publications in various national and international journals and 20 presentations in international and national conference. Sir is the co-guide of 3 ongoing PhD of an ICMR projects in collaboration with other departments and had been a guide for 6 MD and ICMR STS projects in the past. So the chairpersons for the session will be introduced by Dr. Suma.
Thank you, Dr. Dinesh. Now, for this session, we have 2 eminent chairpersons. First is Dr. Surajit Khattak. He's professor and head of department, AIMS, Jodhpur. Sir is a dedicated, resourceful teacher with 26 years of experience at the prestigious Armed Forces Medical College, Pune. He's a graduate and postgraduate of AFMC, Pune. He's a recipient of Army Commander Western Command Commendation in 2009 for outstanding and exemplary work done as professor and head anatomy at the Army College of Medical Sciences, New Delhi. He has authored 2 books in anatomy, has numerous international and international papers to his credit, including the PubMed publications. He has 25 research papers in national and international conferences and 34 international papers published, 15 national, editorials and forewords for books 5, and 2 patents were awarded to him. Our welcome, sir, to the session.
Thank you, ma'am. It was indeed, I was looking forward to this CME for a long time, and I've been in touch with Dr. Yogesh. It's been nice to see this being taking shape, and I would definitely look forward in future also to attend it offline, because Pondicherry, of course, is a place very close to my heart, and of course, this particular subject is very, very interesting and is the most upcoming field in anatomy. And as Dr. Sharif rightly pointed out, most of that, I mean, almost all anatomists must know, because this is one of the most, microscope is our most important tool. And so micrometry and digital image analysis is the in thing, especially for the young anatomists. Thank you, ma'am.
Thank you for the kind words, sir. We are honored to have you. And our next chairperson is Dr. Aruna S., MBBS, MS Anatomy, Professor and Head, Department of Anatomy, Indira Gandhi Medical College and Research Institute, Puducherry. Ma'am is a graduate of Madurai Medical College, 1991, and MS Anatomy from Madras Medical College in 1998. She has profound teaching experience of 18 years. Editor for Southeast Asian edition of Clinical Oriented Anatomy by Keith and Moore. Her areas of interest are embryology and neuroanatomy. Ma'am has 14 papers in international and national index journals and numerous presentations in various conferences. She also has an Cooper Best Scientific Paper Award, which was given twice to her. She has guided multiple undergraduate students for scientific presentations and organized many CMEs and workshops and is a well-known figure. Sorry. We welcome you, ma'am, to the session.
Thank you, Suma. It is an honor to chair up a session in JITMER. I'm very happy to have been invited to do this. Micrometry, altogether a new dimension to anatomy. So I'm very much looking forward for the CME. Thank you so much.
Thank you, ma'am. Glad to have you here. Now over to Dr. Dinesh.
Now I request Dr. Gladwin sir to take over the session. Please, sir.
Thank you, Dr. Dinesh. Can you see the screen now? Can you hear me, Dr. Dinesh?
Yes, sir. Yes, sir.
Thank you.
It is both visible and audible.
Fine. Thank you. I thank the organizing team as well as the chairpersons for giving me this opportunity for a session in this Micrometry CME. So this session will be about the basics of ImageJ, the installation part, as well as the system requirements. So it will be for another 20 to 25 minutes. So we had two three extensive lectures with in-depth information about the various softwares available. But this session will touch upon the very basics about the installation and system requirements and few other basic things about ImageJ software. Before we start, I request the audience to be ready with some sample image for practice for not only for this session, for the next 2 or 3 sessions also. And if you're using a mobile or laptop, please do check for adequate power backup, as well as please remain connected if there are any network issues. So to begin with this ImageJ software. So this is an image processing and analysis program software. It works in Java platform. So that's why the name ImageJ. Most of us been introduced to this ImageJ earlier, but this is an open access software. So anybody can download, it is free to use, and there is no copyright protection involved in this ImageJ software. So this particular software works in both Mac platform as well as Windows, as well as Linux operating systems. And it is compatible with almost all image formats, to name a few, TIFF, JPEG, DICOM, PNG, and BMP formats. So this has been already informed to you in the earlier sessions. And to introduce to you, it also supports tags that are images which have a 3-dimensional voxel platforms. So like images like confocal images, where you have optical sectioning in Z-axis also. And also there can be some things like hyperstacks. So where we call it as time-lapse images, as well as images that are captured by using different wavelengths. So these are series of images that share a single window. And with the help of this ImageJ, it can calculate area and pixel value statistics of user-defined selection area. So this can also measure distances and angles. So that will be demonstrated in the coming sessions. And it can create density histograms and line profile plots if needed. So this ImageJ was started in the year 1987 by Rasband. So he started in the name of Image. So that is associated with National Institute of Health and US-based organization. Over a period of time, so they combined with this Java platform to name it as ImageJ. So from this NaH image to ImageJ, so it's been almost 35 plus years since it is been in use. So there are various formats available. The basic format, it was initially called as ImageJ1, and nowadays we do have ImageJ2 and not many updates of this ImageJ platforms. So now it's been almost 30 plus years. So people have used this ImageJ and they keep developing this ImageJ software as per the requirement. Now, since it is available in this open access public domain, so anybody can download and run the program for any purpose. So there is a freedom to run the program. And you do have freedom to study how the program works and also change it to some of the things you can change to suit whatever the need as per your requirement. And also you can redistribute these copies so that you can help your friends and then neighbors. So no need to worry about this copyright protection. So to begin with the installation of this software, so in the user manual, we have given some links where you can directly click that link and then download the software. But what we usually do, so we just start Googling it. So when you just type these keywords in Google ImageJ download, so you will land up in this particular page. So download ImageJ. So when you click this link, the download ImageJ, so you'll be taken to another window called as download with so many versions available. So since this platform works with Java, so either your system might be having a Java platform already installed in your system, or it may not have been installed. In case if your system has an already installed Java platform, so you can click on this zip archive, so where it will come without this Java platform. So you can use this particular link to download this ImageJ. So in case your system has a pre-installed Java. So this download will be downloaded without Java. In case you don't have a Java pre-installed in your systems, so you can use any of these links depending upon the operating system which you are using. In case if you are using this Mac operating system, so you can download, and in case if you are using this M1 ARM in Mac, so you can download this particular link. And people like most of us will be using this Windows platform, Windows operating system. So you can download this 64-bit Java ImageJ bundle with Java. So this particular link you can download. So this is with Java, right? So depending upon the need, you can choose so which one to be installed. So this entire package comes with a zip format. So when you click and then download, so you will get this particular thing. So there'll be a download. So it will be in a zip bundle. So after completing this download, so as usual, like when you open any particular zip bundle, so you can, it will prompt for some tools. So you can just click open. So you will land up in this particular window where you can extract to another folder, that is the ImageJ folder. So by just clicking this extract, so the bundle files get extracted and all the files will be available in a particular folder called as ImageJ. So you can just choose the location where you want to download and then check the downloadable location and the folder. And you can also verify all these folders which are available after successful download of this particular file. So you'll land up like this. So you'll have some JRE files which are not very significant. You will have some folders called as lookup tables that are some software enabled with this ImageJ, and you have separate folders for macros and plugins. And this is the run command that comes with this ImageJ logo, right? So you don't have to really worry about these folders that are downloaded during this ImageJ installations. So what you need to do is just click on this ImageJ logo. So automatically the ImageJ software gets installed in your system. But before doing this, please also make sure sometimes if you are running an antivirus, so you might have to pause this antivirus to download this particular, to install this particular software. So once you choose this ImageJ icon and then click install, so this particular software gets installed within seconds. So there is no complexity involved in this installation. It will be a very simple, straightforward software. So this is the only window you will be able to see after successful installation, right? So just I'm just repeating whatever I have told. So go to Google, just type ImageJ download. So you'll get a download window available. Depending upon your system requirement, you can just download this ImageJ either bundled with Java or without Java. So it will come as a zip file, and then you can just extract the folders and you will land up in this ImageJ folder page. where you will be able to see this particular icon, that is the ImageJ icon. And once you install or once you double click this ImageJ icon, so you can see this particular software installed in your system, right? So this is the software, nothing much in that, right? Now, in case if you have any troubles during installations, there are instructions available along with this download window. So you can see this one, there are some instructions available for Windows, and there are also some instructions available for Mac platforms. So by clicking on these instructions, so you will have the troubleshooting page also. So in case if you have some issues with the installation, so you can just go through these instructions, and it all basically self-explanatory. You can just troubleshoot and then install it. A word of caution, if you're using a Mac OS, when you run for the first time the ImageJ in a Mac OS, so you can't open that ImageJ file, because sometimes it will prompt an error message stating that it is from an unidentified developer, right? So this can be bypassed by right-clicking on ImageJ app and then click open, right? So if you are using a Mac OS, so this is how it appears. So this is the icon, ImageJ icon. You right-click this ImageJ icon, and then you land up in this particular drop-down menu where you can click open, so that this particular software gets installed like this in case of Mac OS, right? So this is just the window you'll be able to see in case of a Mac OS after successful installation. So you can see the menu bar as well as toolbar after installing in Mac OS, right? Fine. You must be wondering, that's all in this ImageJ software? Is it successful? And you can just cross-check. So I can just show you one particular user interface, and you'll be able to recognize that particular user interface. Before that, you must be wondering this ImageJ, whether there is any other main work area. Unlike most of the image processing programs, ImageJ does not have a main work area. There won't be any any white window or blank window available along with the software. You will have only the menu bar and the toolbars. So ImageJ main window is quite simple, containing only a menu bar with menu commands and a toolbar along with a status and a progress bar, which I'll be showing in the subsequent slides. So any images, so whatever you open through this ImageJ software, or any table which you have derived through this ImageJ software, will be available as a different window. So it will be available as a separate window apart from this menu and then toolbars. So measurements and results are displayed in results table separately, right? So now most of us will be familiar with this particular interface. I think most of you will be able to recognize. So this is the interface for this software Paint. So most of us would have used actually. So now if you see this Paint user interface, you will have this menu and toolbar here. So this is the menu and the toolbar, and this is the work area. So any images you open through this Paint software, you will have these images here, and this we call it as a main work area. But see to that in ImageJ, so you won't have this main work area. What you can see is only the menu bar and the toolbar. So this is how the user interface appears in case of ImageJ. So you can see the main window of this ImageJ, so which has a menu bar like this. So where you can see some of the tools available, like tabs like File, Edit, Image, Process, Analyze, and etc. And you will also come across a toolbar. So where you have different tools for morphometric analysis of this image. And you do have a status bar here. So whenever you process, whenever you try to magnify and do some work with any kind of images, you will come across a status bar here. And you will also land up in something called as a progress bar. So this is how an ImageJ user software interface looks like. So you'll have a menu bar here, a toolbar with all the tools available, and you will have a status bar and a progress bar, right? So nothing much. And the same thing in case of a Mac OS also, you will come across these tools. Now, how do we open an image by using this ImageJ software? So when you open this ImageJ window, so the first tab will be File. And when you click this file, you will have a drop down where you have New, Open, Open Next, Open Samples, etc. Right? So as in case of any softwares like Microsoft Word or Microsoft Excel, so when you click open, so you will be prompted to locate the particular file location. So when you just click that particular image file, so you will be able to open that particular image. So whatever there in this file, it will be all like basics of file operating system. So like opening, saving, and creating new images, right? And there are also other features available under this file menu to just open a new image or to create an image when you want to draw some diagrams or you want to do a hyperstack image. So all these things will be available under this file menu. And also you do come across some of the open samples which are also available with this software. So this open software includes like you have some images in JPEG format and some images in TIFF format and some stacked images and also some hyperstack images. So you can just choose any one of these images and you can have a look at it, how it works and how it looks also. So now this is an example. I've just clicked this file and then I have opened one particular image, right? So as soon as you open this particular image, so this window will become active. This is called as an active window in case of an image J. And the details about this particular image will be shown on the top end of this particular image. So you can see this one. So the name of that particular image and how much is it magnified and what is the pixel and what is the RGB values, the size, etc. will be shown in the top end of this particular image. And similarly, when you look at the status bar, when you keep the cursor in any part of this particular image, the coordinates will be seen in this status bar. So what is the X coordinate and what is the Y coordinate? So that particular information will be shown on the status bar. And maybe you would have heard about this RGB value. What is the R value, G value, and B value? Red, green, blue. So these values also will be shown in this particular status bar. So you can see here in this particular example, so you come across these values 233, 173, and 227, right? So you can just check and open white image and black image, and then you can just check how these values keep changing. And similarly, you can just move the cursor across the images, and then also you can just follow it up how the X and Y coordinates keeps changing, right? So this you can just try whenever you find time. Now, so all image operations are performed in an active window. Suppose in case like you open some 2-3 pictures, so whichever the picture in the front part, like in the front window, that we call it as an active window. So always in that active window, the title bar will be highlighted. And also you can zoom in and then zoom out of these images up to a magnification of 32, 1:32 ratio, right? So by using some simple commands, Control plus or Control minus, you can just, or scroll, you can just zoom in or zoom out. So this is an example I can just showing you, like there are two pictures now, and this is the front pictures and this is active. So you can see the title highlighted in this active window, right? So when you're working on this 2-3 images, then you might get a little bit confused. So which X and Y coordinates it is showing, or if you move the cursor to that particular image, that image will become active. So you can just see from this highlighted active window that is seen in this top end of this particular images, right? Fine. So this is about active window when you're using more than one images. Now, when you right click any image while you're using this image date tools, there are several drop downs available where you can rename that particular image or duplicate that particular image. The golden rule for any image processing will be, before you start meddling with any particular image, you need to duplicate or you need to have a separate copy of the original image, right? So that is available in this particular commands that are available in right click. So you have something called as rename, duplicate, etc., where you can rename also, where you can duplicate also, and then save separately, right? So these are few things which are available after installation and under this file command. Now, few word about this image coordinates, which we have discussed in the earlier presentations also, and also in subsequent presentations. Now we all know like these are the X and Y coordinates. So whenever you keep the cursor across these images, you will be able to follow this X coordinate and also Y coordinates, right? And previous presentation, the chairpersons also, they were mentioning about stacking. So when you have images obtained from confocals, so you will have optical sectioning of that particular image. So you have images in a three-dimensional format. Sir was mentioning about voxels. So now these are called as optical sections, and these optical sections, we call it as a under z-axis. So these are z coordinates. So in addition to x coordinate and y coordinate, so we have one more coordinate that is called as z coordinate. So these are available in case of a confocal image series. So when you use a confocal image, so when you open under this image J series, it will open as stacks, and it will have x coordinate, y coordinate, as well as z coordinate. Now, some of you have been wondering, like, if there is an overlap of nuclei or something like that. Suppose if the image has been obtained from this confocal image series, there is a possibility of optical sectioning of images. Now, this is an example which I was telling you about Z stacking. So these are like different optical slices. So we have 6 optical slices. These optical slices will be combined to form one active window, and so you'll have Z coordinates. So you have 6 Z coordinates for this particular image. Now, so these are called as stacks, and there are something called as hyperstacks. So nowadays we do have other dimensions also. So when you do some confocal imaging or something like a fluorescent imaging, you do capture images under different wavelengths. So you will capture under blue light, or sometimes you capture under green light, you capture under red light. So you have different channels. So 3 channels are available. These are captured with different wavelengths. So these are called as C coordinates or channel coordinates. And then lastly, something called as T coordinates. It is like time-lapse images. So in this picture, I can just show you. So what do you mean by multiple channel images? Now, so this is a confocal image. First, this particular image is taken by means of this blue light. So blue light is captured. So you can see channel 1, right? So then later you'll be capturing the same image with the green emission filters. Like, so you can see only the green ones. So now you have green as well as blue highlighted. So the same picture. So the first one was only the blue images. And now you have, along with this one, the green light. And when you go for the channel 3, so you'll have this red emissions captured also. So these are like 3 channel images. So you have blue, green, and then red. So these are called as hyperstack images, which also can be processed by means of this image J software, right? So you can see this 3 channels enabled for this red, blue, and then green. And similarly, sometimes we do have this time-lapse images. So the images which are captured over a period of time. So this will be the 5th coordinate. We call it as T. So for example, these are cell images which are captured at 0 minutes, and then after 10 minutes, 20 minutes, likewise, after some periodic intervals, right? So these are called as time-lapse images or T coordinates, right? So now in case of image J, so these hyperstacks are shown in one particular window, that is one active window, but you will have this scroll bars that are seen below this image. Now, as I mentioned, this C stands for channels. So when you scroll this C, so you'll have some channels like different wavelengths from blue, green, and red. And similarly, so when you have this Z scroll bar, when you scroll through this Z coordinate, so it will be the slices, it will be the optical slices. And similarly, there'll be one more coordinate that is called as T. So these are images captured over a period of time. So these images are called as hyperstack images. So from basic images, that is the 2D images, X and Y coordinates, we can analyze 3D images also, X, Y, and Z coordinates, and also hyperstack images, X, Y, Z, as well as channel images, as well as time-lapse images. So these are added advantages using this image J software, right? So before I wind up, again, that emphasis on the image file types. So Dr. Sharif sir was telling, so always we need to use TIFF format for this image processing as far as possible. So TIFF format is called as tagged image file format. And other formats which are commonly used are this JPEG format. It is called as Joint Photographic Experts Group format. So nowadays they also call it as JPG format. And these are DICOM images which are often used to analyze the CT and MRI pictures. So these are called as Digital Imaging and Communications in Medicines. These are the DICOM images. Now, why we need to use this TIFF image format? So TIFF images create very large file sizes. So this we are telling, whenever you capture an image under a microscope, you need to save as a TIFF format. Though the file sizes are large, so these images are stored as an uncompressed, thus contain a lot of detailed image data, which is why the files are too big. Now, TIFFs also can be extremely flexible in terms of color. So they can be grayscale images or CYMK format or RGB format. So these are some added advantage while using this TIFF images, right? And this is also the most common file type which are used in a photo software. All other formats like JPEG and PNG formats, they use something called as a compressed format. So when we compress the image, so when we save the image in a JPEG format, we will lose some information about that particular image. So most digital cameras use this JPEG format. So we can't resolve much from this JPEG images, right? So JPEG is a compressed way. So we call it as a lossy compression. So whenever an image is compressed, there are two types of compression. One is a lossy compression and a lossless compression. So this JPEG format uses something called as a lossy compression, where we lose the image data, right? And so we can reserve something JPEG images only for something like digital images for this one digital cameras. And when you have this micrometry work, so please use only this TIFF format. This is an example I wanted to show you, like when you compress images, what happens. So when you start compressing images, you know that file size keeps decreasing. So as the file size decreases, you will be able to see this one, the pixels appearing, right? So you can see this fine image quality in this compressed files. So this is called as a lossy compression, which the JPEG format uses. But in case of TIFF, so it uses something called as a lossless compression. The file size will be larger, but you will get lot many information, right? And so these are the DICOM images, the digital imaging and communications in medicine. So these are images by which you can analyze this MRI and then CT scan images. These are serial sections. So this will be taken like a stacked images. So whenever you open a DICOM images by using this image-based software, it will open as stacks and also hyperstacks, right? So that is also an added advantage of this image-based software, right? So as such, to sum up whatever this installation part, it will be very simple. With few clicks, you will be able to install this image-based software. It works in any platform, Windows, Mac, or Linux platform. So this is the working window of this image-based software. You will have a menu command, toolbar command, status bar, as well as this process bar. So these are the few things available in case of this image-based window, right? So to sum up, this is an open access. You can just go to this NIH link, National Institute of Health. With few clicks, you can just download and then install this image-based software, right? So these are the various features available in this image-based user interface. And you have learned a few things about this coordinate. I wanted to re-emphasize because henceforth the two sessions will be on the demonstrations. So the X coordinate, the Y coordinate, the Z stacking, the C for channels of images captured for different wavelengths, and T for this time-lapse images. And to wind up the file types, so always use TIFF images whenever you want to process any images for micrometry work, right? So thank you for patiently listening. If you have any queries, I'll be able to take up one or two questions. So I thank the chairpersons as well as the organizing team for giving me this opportunity.
Thank you very much for the wonderful session, sir. The session gave wonderful insights into installation and basic tool options of image software. And I hope at the end of the session, the audience will be clear about how to install them and how to use according to their settings. So the questions also, in the due course of questions we had in the chat box, like, are there any special type of image that can be opened? But I think at the end of the session, they would have got clarified like about the TIFF image, how to use, what is the difference between TIFF and the JPEG image and so. And one person has asked about demonstrate the paint options, and it will be done in the subsequent sessions. And one question is how to select the image, sir. How to select?
Yeah. So selection of the image can be done. So there will be a file command available here. You can see this one, file tab. So when you click this file tab, so you'll have a drop down. So there you will have something like an open new, open recent something. So if you click this open command, so then you will be getting a prompt to locate the place where you have stored that particular image. For example, in your document, this one, if you have a folder of all these images, you can just click those location and then click that particular image. It will get automatically open with this images software. It's just like how do you locate this Microsoft Word, Microsoft Excel file. The same way you can just locate this any image file through this images software.
Yes, thank you, sir. Now I request the chairperson, Dr. Surjit Katak sir, to give his opinion and ask questions if any. Welcome, sir.
Thank you, Dr. Gladwin. It was indeed a very, very lucid presentation. And it was, I was very saying to see how easy it is to download and install this particular software, which is freely accessible. So I got a small query. What is the difference between image J and Fiji?
I think that was explained in the earlier session, sir. Image J is a basic version. That was the one, so they have merged image and Java platform. So they made it as a image J. And Fiji will have all the plugins available along with this particular software. Plugins in the sense there will be some additional tools available as a software package. So in image J, all the basic plugins are available. So when you go for this advanced micrometric imagings and measurements, in case if you need some additional tools, you need to download some plugins or macros in additions which are available with the software. In case of Fiji, so almost all the plugins are ready-madely available. That's a difference, sir. But for this basic work, that's not much of difference with this image J2 and Fiji.
And so these images can be, it can be printed also. This, we can use this software to print also.
Yes, sir. Like so, once you are done with this image processing, if you want,
yeah,
definitely you can do that.
Thank you, Dr. Gladwin. It was indeed very nice to come to know about this particular software. Thank you so much.
Thank you, sir. Thank you.
Thank you, Dr. Katak sir. Now we call upon Dr. Aruna ma'am to ask questions if any and give her opinion about the session. Welcome, ma'am.
Good morning to one and all. First, I thank the organizer team for giving me this opportunity to chair a session which was given by my UG junior, Dr. Gladwin. So we both know very well, before I joined this institution itself, I know Gladwin very well. He is an excellent teacher, a very good, simple explanation he usually gives. So no wonder he did this presentation also in an excellent manner. Very simple explanations, very simple English language, which can be understood by everybody. And he has explained with the screen. So demonstration was very, very nice. So the micrometry, actually, it's a new dimension to anatomy. When we did post-graduation in anatomy, we were not given much emphasis on histology. We'll be concentrating only on gross anatomy, osteology and other things. Histology is done only for the sake of passing the exam. Now that has taken a new dimension with this J image technique and micrometry, and it has grown so far. I'm so surprised to see that anatomy has got a new dimension altogether. I'm so happy that JIPMER has taken an initiative to introduce this to all the other people, the young anatomists, because gross anatomy is slowly fading away. With the dirt of cadavers, it's slowly fading away. So this will give a new opportunity to the anatomist to excel in the anatomic field. Besides gross anatomy, they can show their excellence in the picture and this micrometry also. So very excellent presentation, Dr. Gladwin. You have explained it in a very simple manner. I think every explanation is given by you and previously by Dr. Sharif and by Dr. Vasudha also, whatever questions they have explained so well. I thank you. Thank you for the opportunity. Please do something like this repeatedly, especially the recent advances in anatomy. This will be very useful for the youngsters. Good luck to Jupma, Dr. Shuma, and their team. Thank you.
Thank you. Thank you. Thank you for your wonderful opinion. Sir, I request Gladwin, a few more questions are there. The first question is, can it be done through mobile?
No, so those some are compatible with this mobile. Always when we do this micrometry part, so always advisable we need to use a system. So yes, plugins and macros usually works better in case of a desktop or like laptop systems. So for this micrometry work, so we need to avoid using through this mobile, and also this capturing images through this mobile and all not advisable.
So can older stain slides can be used for micrometry?
See, micrometry is like just using some of the tools to analyze the image. So any image can be used, even if the slides are old, it's fine. So we need to have a good microscope with an attached digital camera or a video, so that you capture the images, and post capturing you need to use these softwares to analyze those images. So for image processing only, we are going to use this particular softwares. For capturing, you need a microscope with an attached digital cameras. For that purpose, so you can use any type of slides, old slides as well as new slides.
Yes, sir, can ImageJ process raw format images?
Yes.
Yes.
It can be used. It can be used.
Yes, sir. What is the accuracy of measurement in ImageJ compared to digital vernier calipers?
It is precise and accurate. That all depends upon the initial calibration. Suppose yes, we capture an image at 10x, so then we need to calibrate first according to this 10x magnification. So we do capture images under 10x, 20x, 40x, and also the 100x. So first we need to calibrate that particular image under which magnification we have captured, and once we calibrate, these measurements are accurate.
So then last question, sir, how you number the images in z-axis when it is split?
So numbering will be done automatically. Suppose when you have this confocal imaging, so when you have a thick specimen, when you want to optically slice, you will be manually assigning how many optical sections you need to take, which how much thickness. Suppose you want to take 10 optical slices, so we will be prefixing that particular 10 optical slicing. So then, so it will be numbered 1, 2, 3, 4, 5, 6, something like that. So the 10 numbers. So that during this image capturing, so you need to process that actually. So you need to assign like how many images to be slide, how many optical sectioning. Similarly, for this time-lapse imaging, after how much period you need a capture. So after every 10 minutes. So something like that, that you have to work before capturing.
Yes, sir. Thank you, sir. So with this, we are concluding the session. It was indeed very wonderful and highly informative. So now I ask the chairpersons to present the certificate to the speaker, Dr. Gladwin, sir. Herewith, I am unveiling the certificate of appreciation to Dr. Gladwin, sir.
Thank you.
Please, sir and ma'am.
Congratulations, Dr. Gladwin.
Thank you, ma'am. Thank you, sir.
Congratulations, Dr. Gladwin.
Yes, sir.
Yes, sir. Now I ask Dr. Suma, ma'am, to present the certificate to Dr. Surjit Katak, sir.
Thank you very much, Dr. Surjit Katak, sir, for sparing your time and acting as a chairperson for this session. Thank you very much, sir.
Thank you. Thank you.
Congratulations, sir. It was nice.
Now I ask Dr. Suma, ma'am, to present the certificate to Dr. Aruna, ma'am.
Thank you very much, Dr. Aruna, ma'am, for sparing your time of the busy schedule and attending the online session and giving you your valuable comments. Thank you very much, ma'am.
Thank you, Dr. Shuma. It was my pleasure.
Thank you.
So we are going to the next session of the day, and the next session will be another one live demonstration. And this live demonstration is entitled as User Interface and Calibration of ImageJ. This live demonstration will be rendered by Dr. Sulochana Saktivel ma'am, Additional Professor, Department of Anatomy, JIPMER. Dr. Sulochana Saktivel ma'am is MBBS, MS Anatomy and PhD. She is currently Additional Professor in the Department of Anatomy, JIPMER, Puducherry. Madam has done her graduation, that is MBBS, from Tirunelveli Medical College, her post-graduation MS Anatomy from Tanjavur Medical College. She has 14 years of experience in undergraduate teaching and 8 years of experience in postgraduate teaching. She has 18 papers in various national and international journals, and she has presented 20 presentations in various international and national conferences. She has been the resource faculty and organizing secretary in 3 workshops. She has received the Best Paper Award in 3 conferences, and she has received the M.M. Cooper Memorial Award for the best presentation once. So we are indeed very grateful to ma'am for accepting the opportunity for presenting in this CME. And now we would like to introduce the chairpersons for this session.
We welcome our respected chairpersons for this session. First is Dr. Rama Devi, MBBS MS Anatomy, MNA MS Anatomy, and PGDBE, Professor in Health, Department of Anatomy, PIMS, Puducherry. Ma'am is a graduate of Medical College Trivandrum and MS from St. John's Medical College, Bengaluru. She has set the Division of Human Genetics with a Clinical Genetics OPT, a counseling section, and a cytogenetics lab in the PIMS, Puducherry. She is a visiting professor observer in the Department of Genetics, University of Minnesota, Minneapolis, and clinical geneticist in the Division of Human Genetics. Ma'am is a technical assessor and expert in genetics NADL and has been teaching to the students of medical, paramedical, nursing, and the science students. Her other credentials are she has organized CME workshops in genetics in the department in more than 80 scientific presentations and conferences, and participated as faculty in more than 60 CMEs, workshops, and seminars. She has over 50 publications in peer-reviewed journals, more than 25 projects in genetics, anatomy, and medical education, and various responsibilities, including that she is a technical expert advisor in genetics NABL, member secretary in various committees, and EC member for various institutes. We welcome you, ma'am. Our next chairperson is Dr. Virendra Kumar Nim, sir, MBBS MS Anatomy, Professor, Department of Anatomy, PIMS, Puducherry. There is MBBS from JIPMER and MS also from JIPMER. He has worked as medical officer for 4 years in Arunachal Pradesh and Andaman and Karaikal. He has worked as faculty in KMC Mangalore, PIMS, and PIMS Puducherry, where he's working till now, and he has guided the MD, MS, and PhD students. Sir has several publications and presented many papers in conferences and chaired many scientific sessions. He's a member of ASI, SOCA, IMA, IMSA, KCA, Tamil Nadu Association of Anatomists, SFP, MS, ISHG. He's examiner and paper setter in several universities and involved in exam-related and other activities of several universities. Sir is also NAC assessor for several universities. Welcome you, sir.
Thank you.
Over to Dr. Dinesh.
Now I would call upon Sulochana Madam to give her session. Here.
She's coming.
So you can put in the system.
Sorry for the inconvenience. We are trying, actually some internet issue is there. So we will get Dr. Sulochana very soon. Please bear for a few minutes. Thank you. So thank you for joining the CME. We are waiting for Dr. Sulochana to log in due to some internet issue we are facing here. There is some server maintenance from the Pondicherry government side, and as this is the Institute of National Importance, we are sitting in JIPMER, we are taking only the government server for the internet as a service provider. So we are facing internet issue due to that one. Today some server maintenance is also going on from the government side. So please bear few minutes. She will log in from the another system. Welcome, Dr. Sulochana. Am I audible there?
Okay, Shah, you are audible. Sorry for the delay. There was some internet connectivity problem here. So in this presentation, we will see how to use the interface and how to do the calibration. So first, we already know about the this is the user interface you will see in the image J. We have the menu bar, tool bar, and the status bar. Okay. First, let us see the menu bar. Here you will find file, edit, image. So here you will find file, edit, image, process, analyze, some plugins, window, and then help. So participants were given some 4 pictures today. It was sent in WhatsApp. So you store them in the folder and let us see how to open the image. So first you will click the file, then go to open. It will open a folder. For example, so this is the image user interface. Here I will click the file and go to open. When I click it, it will open a folder. So my images are stored in the desktop. So these are the 4 images that were sent to the participants. Here I will open the image, click open. So the image will open here. So now you can see the image. So this is how we open an image in the image. So go to file, open. It will open a folder. Wherever the image is saved, you go there and open that image. Open that particular image. I have opened this image histological picture here. Okay. If the picture is not very good, you can adjust the brightness here itself. You can go to the image, you can do adjust, and here you can select the brightness or the contrast, and you can minimize or maximize the brightness. So that is how we will open the image. So next in the edit, this is similar to any other program in the Microsoft. You can undo the picture or cut, copy or paste, all those things we can do. Next is the image we have already seen, the process. So during this presentation, we will learn how to add noise in the picture that we will see when we are setting the calibration. So next, we'll see how to analyze. In this analyze drop-down menu will come. Here we will set scale. Set scale is used to calibrate the image J. So next is the toolbar. Here you can see various tools which will be useful in selection or drawings and everything you can do in the using this toolbars. You will see one by one. Okay. First one is the rectangular tool. So side by side participants can open the image J and you can draw a rectangle on the picture like this. So this we can use a rectangle. If you press hard on this, press long press, if you do long press means it will open. This is not opening yet. Sorry for a problem here. The image is hung up here.
We are again sorry for the interruption in the session due to the huge issue regarding the internet, and sometimes our systems are not compatible with the softwares. As it is the free software, the system developers sometimes they may not provide us to work with this thing. Ma'am will continue the session. Please, Dr. Sulochana, ma'am.
So let us continue with the picture, the image. That image was not opening in that computer. Okay, let us see only the PPT. Participants can follow me. So next, in the process, we can add a noise that we will learn later. The analyze is the other one. In the drop-down menu, you will find the set scale. So this is what we will be using here today to calibrate the image. So these are the various tools in the toolbar. First one is the rectangular one. Okay. So if you long press this rectangle, you will find 3 more rectangles that come. That is a rounded rectangle and a rotated rectangle. So when you click on this rectangle, if you double click it, the options will come where you can change the width of the rectangle, or for a rounded rectangle, you can say how much curve you need for the rectangle. So next one is the oval tool. So when you long press that one, you will get a oval selection, elliptical selection, and a selection brush tool. Okay. So oval will give you a particular oval shaped structure where you can do it on the selection, and the elliptical one gives a elliptical picture. Next one is a polygon selection tool, where if you want a different size of an image, there you can do the polygonal selection tool. The next one is a freehand selection. So for example, I have given you the picture of the kidney. So if you want to select only the glomeruli, you can hand draw this glomeruli over the picture. So this freehand selection tool, next one is a straight line. In the straight line selection, you will have a straight line, segmented line, and also the freehand line. And then an arrow tool also will be there. So next we have the angle tool. If you want to measure any angle between 2 particular structures, you can use this angle tool. Next one is the point tool. So point tool, there is a single point tool and also multi-point tool. So point tool can be used to count the number of cells in a particular slide. So you choose a point tool, it will give only one point. So if you choose a multi-point tool, you can add points to all the cells which are present inside the image. So next one, the text tool. So if you click on the text tool, it will give us a text box in the image. And if you right click on this text A, it will give the various options to change your font size or to change the font also. And next is the magnifying glass. So this magnifying glass is used to zoom in or zoom out. Okay. Next one is the scrolling tool. Scrolling tool, when you are using a large image which is not fitting into the image, into that window, image window, there you can use to, instead of using the mouse, you can use this scrolling tool to scroll faster through the image. So next one is the label tool. Okay. So in the text tool also you can use, like this particular A is also used for labeling. You can use a label tool which will give an point so that you can label it in a sum. If it is a small nuclei you want to label, means you can use this label tool to label the particular cell or a neuron. Then when you click this red arrow here, more tools option will be coming. Like you have the startup macros, arrow labeling tools, built-in tools, drawing tools. So mostly you'll be using the drawing tools, arrow labeling tools in the built-in tools.
Thank you, sir. Thank you.
Now let us go to the set scale.
All guidance and everything. Our department setup करने में तुझे तो पता है हमारा department कैसा है. इसके बराबर या इससे बेहतर बनाना. ठीक है. That should be your aim. ठीक है. कि इस aim को हम कैसे हरा सकते हैं. ठीक है. So that you can do because you have seen this.
For doing the set scale to calibrate the image, we need micrometers. There are two types of micrometers. One is a stage micrometer and the ocular micrometer, which has been already described by our ma'am. So for this particular calibration, we need a stage micrometer. This ocular micrometer is a circular one, which has some divisions marked on this one. So this ocular micrometer is actually placed within the eyepiece. So when you are viewing this objective 10x or 40x, both will remain the same only. So we cannot use this ocular micrometer for calibrating the image. We need a stage micrometer. So stage micrometer will have scale of 1 millimeter. The small line here is a 1 millimeter. It will have 10 large divisions. 10 large divisions will be there. So this is how this stage micrometer will look like. So this whole distance is 1 millimeter. This is divided into 10 large divisions and 20 small divisions and 100 smaller divisions. Okay. So this 1 millimeter is equal to 1000 micrometer. Okay. So 0.1 millimeter is 100 micrometer and 0.01 millimeter is 10 micrometers. So our large unit here will be 100 microns and the smallest unit will be 10 microns. The small one will be 10 microns. So this is a stage micrometer taken under the objective 40. So this large division is 100 microns. The smaller division will be 50 microns. The smallest one will be 10 microns. So task for us to now participants has to open an image by choosing the file.
Ma'am, it is not audible. Sulochana ma'am, it is not audible.
I'm very sorry for this. Is it audible now?
Yes.
Okay. So now we come to the task how to set the scale in the image. So let us open this image and go to the file and open the image that was sent to the participants, that is the objective 10x. So this is the objective 10x, which is micrometer.
Can you stop share and new share? New share.
Nice. Okay, let us now start open a file. So I will open the stage micrometer taken an objective 10x. So now we know we are familiar with the straight line tool. Take a straight line and draw a line from the end, the whole of the 1 millimeter stage micrometer. After you have drawn a line, go to analyze, set scale, and you will find some of the distance in pixels. We'll have a known distance and then pixel aspect ratio and the unit of length. Okay. Now the known distance means we already know what type of measurement we have in this one. This is a stage micrometer of 1 millimeter, or else it is a 1000 micrometer. So in the known distance, we will write 1000. So pixel aspect ratio will not do anything. We'll leave it as such, and let us come to the unit of length. So we know the unit as a micron. So we'll add micron in the unit of length. So next there is one box which calls as global. Okay. If you click that global, so whatever image we are going to use hereafter, this scale will be used in all the images, all the consecutive images we are going to analyze. Okay. Then you click OK. Now we have set the scale. So now we have set the scale. So I'll repeat again. So what you do is go to File, open the 10x objective of the stage micrometer and open it. So when you open, draw a straight line, that is choose the straight line tool and draw a line from the beginning to end of this 1 millimeter divisions. Okay, after that go to analyze, set scale, and here you will find distance in pixels, known distance, aspect ratio, and unit of length. We already know the distance of 1 millimeter, that is 1000 microns. So we write it as 1000 in the known distance. And then the unit of length is micron. So you put a micron. Okay. If you click the global, this scale will be used in the consecutive images we are going to analyze. And then click OK. Now let's go to the file again and open another image. So participants were sent a image of kidney. So open the 10x image. Okay. Now we can see the measure any of the glomeruli seen here or any other tubule seen here. Let us take this glomeruli now. Draw a straight line from one corner to another. Okay. And then go to analyze and then measure. So here it will open a result box. Here the length of the glomeruli here is 76 microns. Okay. Or you can again measure from here to above and then analyze and measure. You will get another result. So the size of this glomeruli is 76 into 94 microns. Okay. Similarly, we can measure the area by using a freehand tool. Let's go to image J again and open the image. Take the freehand tool and draw on the glomeruli. Okay. Then again, go to analyze and measure. Sorry. So like that, you can calculate the area of the particular. So next, you can count the number of glomeruli also, number of cells also. So this we have already. So let us count the. So here we have to use the multi-point tool. So if you click on the glomeruli, it will write 1, 2, 3, like that. It will go on using this multi-point tool to count the number of glomeruli in the particular slide. Okay. So let us begin. I will repeat the set scale section again. First, you open the image J, open the file, and go to open. It will open the new folder there. So what you have to do is, first you have to open the objective 10x, that stage micrometer where you have taken a picture with the objective 10. Okay. So this will open here. So what you can do here is, this image is a little bit darker. You can adjust the brightness of the image also. Adjust brightness. So like that, you can adjust the brightness and the so now we have to go to the straight line. Okay. So draw a line from the beginning to the end. So approximately it is coming to 1002. So you can, if you click on this one, so you're drawing a line from here to here, go to analyze, set scale. Okay. So here there are 4 units: the distance in pixels, known distance, pixel aspect ratio, unit of length. So now the known distance is coming as 1002.53 because we have already set the scale to 1000. Okay. So what you have to do is click to remove the scale. So let us start from here. You have drawn a line from the beginning to the end. Just the line. Go to analyze, set scale, and then here the known distance is 1000, right? And the unit of length is micron. Okay. Apply global and then click OK. Okay. So we are setting the scale now. So this is a 1 millimeter, that is equal to 1000 micrometer. Now let us open an image. Please open the kidney in 10x. So this is how. Now click the straight line and draw a line. If you want to measure the glomeruli, draw one end. Draw a line from across this glomeruli. Go to analyze and then measure. I'm so sorry, it is not working again. So let us do the same thing with a 40x objective, that is stage micrometer taken under the objective 40x. So this is the image here. Let us go to the straight line, draw a line from this one end of the larger division to the other end. Okay. So this distance, we know it as 100 microns. Okay. So go to analyze, set scale, and in this one known distance, you have to write it as 100 microns. The unit of length is a micron. Apply global and hit OK. Again, you have to go to the file, open the new image, the image which was taken under objective 40x. Now you can draw a straight line. You can measure the size of the tubule here. And you have to analyze the the result box will open, but particular in this image it is not opening. So here you can see the result. So this ninth one is the last one we have done. It is the 50 microns. The length of this tubule is 50 microns. Actually, you can draw another one from here to here and analyze and measure. The next one will come. The 10th measurement is 185 microns. So next thing we can do is we can see the area of the. So again, let us open the image in 40x, or you can open the 10x image. Here, let us measure the area of the glomeruli. So take the freehand tool and draw a line over the. If you click on the image, that last measurement, that line we have drawn, no, that will disappear. Okay. So click on the freehand tool, draw a line over the glomeruli. Okay. And then again, and the measure. So here, what you can here see is the area. Okay. So this gives the area of the particular glomeruli we have chosen. Okay. So when you are drawing a rectangle, you draw a rectangle like this. Right? Okay. So right click on the rectangle, add to overlay, means if the rectangle will stay in its place. So otherwise, you draw a rectangle and click it on the side, the rectangle will disappear. So whenever you want a particular measurement or marking to done, you have to right click and add to overlay. Particularly, it will stay in that place. You can go to edit and undo. So multi-point tool can be used to count the number of glomeruli in a particular section. So it will give, you can see the number also 1, 2, 3, and this is 4, and this is 5, and this is 6, 7, 8, etc. So I will repeat all the user interface tools here. We have a rectangular tool to select a particular area. Same way, oval tool and elliptical tool can be used. If you want the irregular shaped structure should be selected, you can use the polygon tool, and you can draw a freehand also, like a glomeruli what we have done. Straight line, you can label it. And the next one is to find the angle, and then we have a multi-point and a single point tool. Then wand tool is there. Wand tool, you can select a particular area. For example, if you open an image, draw a line, draw a point here. Just select this white area to select the areas which have a uniform color. So this one tool, which will be explained later in the next session. Next one is the text tool. So enter the text. So if you right click on it, you can increase the font size. Okay. You can change the color of the font kidney from here. You can pick any color or just go there. You will come to that later. So let's stick the color picker. So this next tool is the color picker tool. You can pick any color from the particular. Next one is the more tools which are seen in the last one. Here you can see the arrow labeling tool, built-in tool, drawing tool, and magic montage, all those things. Here you will be using a drawing tool and the built-in tools. Another tool that is important is the labeling tool. You can label it like this. So this, it will open a small box over there where you can write it as. So today we have learned about how to set the scale in the image J. So now we know about what are all the tools available on the interface and how to set the scale using the 10x objective and 40x objective. So the most important thing you have to know is when you are taking a picture of a stage microtome under 10x objective, the picture should be taken from the same microscope under the 10x objective. So you want to to set the scale for that particular image. The 10x objective image of the stage micrometer and the 10x image of the particular histological picture should be used. If you want to set scale for a 40x objective stage micrometer, you have to take the picture taken from the same microscope with the objective 40x. So that is how we have to set the scale. Okay. Thank you.
Thank you for the wonderful presentation, ma'am. Now, I request the chairperson, Dr. Rama Devi, madam, to ask
Dinesh sir, you
questions
are not
and also
audible.
give her valuable opinion about the sessions.
Dinesh sir, you are not audible, sir.
Dr. Yogesh is audible. He is audible.
Question 6. First chapter.
Hello, am I audible?
Yes.
It was a wonderful session by Sulochana because we started off the day with what is micrometry. Am I audible?
Yes, madam.
Yeah.
Yes, ma'am.
We started off with micrometry and then we had the next speaker talking about image analysis, which image analysis, the software to be installed. And then we were all curious to know as to how you use the software and do your measurements. So Sulojana took us through a beautiful tool, a beautiful session of how to do the analysis step by step in spite of the technical glitches that we had. I don't think that was a break in our following up the procedure, getting to know as to how to use the tool, because multiple times she explained and the different tools also she did. I'm sure that all the participants would now be excited to do it by themselves, because already Gladwin has shown you how to install the software. I'm sure that would be the thing that all of us would be doing today evening at home. And now Sulochana has shown us how to get a slide. So the next procedure which all of us would be doing is try to procure an image and keep for you for the homework to be done by ourselves today. It was a wonderful experience and a good presentation by Dr. Sulochana. Thanks a lot. At the same time, I thank the organizers for letting me be a part of the program and to follow up as to what is happening today. All the best for the subsequent sessions as well.
Thank you, ma'am. Thank you. Now I request Dr. V.K. Nimsar to give his opinion and also give some suggestions regarding the session.
It was a it was a nice session. The first, it was a theoretical aspect, then the how to download and set all these things, and then Sulochana has done how to use the micrometry. Rest of the thing Dr. Rema already told. This session remind me 44 years back, I used ocular micrometer and stage micrometer when I was doing BSc. There we use to measure the stomata, diameters, all these things, measurements. Now this thing, that was just manual. Now it is, everything become digital. That is the progress. All the best for this thing. Thank you for the opportunity.
Thank you, sir. Now I will ask only 2 or 3 questions from the audience. Remaining questions will be answered and it will be sent to you by the speaker in a short while. So first question, can we calculate the area of placenta by opening the gross image of the placenta in the app and changing the scale units to centimeter?
Yes, you can I showed how to measure the area of the glomeruli, isn't it? So you can choose the freehand tool and draw a line, circle around that area you want to measure, and you go to analyze and click on the measure. It will give the area of that particular section you have selected in the image. So you can do it in a gross image also, you can do it in a histological image also.
Then second question, from where can we get the ocular micrometer which you showed in the beginning?
Ocular micrometer is available anywhere and in any scientific labs actually. The stage micrometer can be bought in the Amazon also. It is around some 300 or 400 rupees only. But we need a stage micrometer only for this image software. Ocular micrometer is of no use actually.
Then how to take the thickness of fibers such as collagen and elastic, which are of varying profile? Do these hinder with the reliability of interpreting?
This you can see in the live demonstration in the next session by Dr. Yogesh.
Yes, ma'am. That's all. So remaining questions will be answered and it will be sent to you in a short while. So with this, we are coming to the end of this session. Now, I request the chairpersons to unveil the certificate to the speaker, Dr. Sulochana Saktivel ma'am.
Congratulations, Sulochana.
Thank you, sir.
Congrats. Beautiful presentation.
Thank you, sir.
Now, I request Dr. Suma ma'am to unveil the certificate to the chairpersons.
Thank you, Dr. Rema ma'am, for spending your valuable time and giving your suggestions and comments for this session. Thank you very much, madam.
It's my pleasure, madam.
Thank you.
Thank you.
Now, the certificate will be awarded to Dr. V.K. Nim sir.
Thank you very much, Dr. Nim sir, for staying throughout the session from morning and like sharing your experience with us. Thank you very much, sir.
Thank you. Actually, I asked Dr. Suma whether it is offline or online. I prefer offline.
Yes, sir, definitely, sir.
Because I want to go visit JIPMER as many times as possible.
Sir, yes, sir.
It's alma matter for me.
Thank you, sir. Thank you, ma'am. So now with this, we will be going on into the next session of the day. That is a live demonstration session, histological image analysis using ImageJ. And this session will be rendered by Dr. Yogesh Ashok Sontake sir, Additional Professor, Department of Anatomy, JIPMER, and currently the organizing secretary for the current workshop and CME. Dr. Yogesh Ashok Sontake doesn't require any introduction, as many of us will be knowing about him through his textbooks and also through various other platforms. He has the educational qualification of MBBS and MD Anatomy, and sir is currently the Additional Professor, Department of Anatomy, Academic Center, JIPMER. Sir has done his undergraduate from Government Medical College, Nagpur, Maharashtra, and post-graduation from Jawaharlal Nehru Medical College, Wardha, Maharashtra. Sir is currently working and has worked upon various immunohistochemistry projects, and for those projects he has used confocal microscopy, and for those projects he has utilized the total grant of 7 lakh rupees. He has a profound expertise in Adobe Illustrator software, and he has conducted various workshops based upon it. And through his textbooks, we all know about how far he will be using for Adobe Illustrator software for medical illustrations and Premiere Pro software for editing educational videos. He has published nearly 49 articles in various national and international journals. And this is the list of books which has been published by him. It starts from the textbook of human histology, human embryology, human anatomy, which is ongoing, principles of clinical genetics, principles of histological techniques, and principles of scientific writing. As you all would have seen one or other of his book, and I don't require to introduce about his textbooks anymore further. So with this introduction, I welcome Dr. Yogesh sir to give the presentation. The chairperson for this session are.
So we welcome the esteemed chairpersons for this session. First is Dr. Brijendra Singh sir, Professor and Head Anatomy and Dean Social Outreach, AIMS, Rishikesh, India. He's President, Anatomical Society of India, Vice President, Anatomical Society of India, UP Chapter, and Vice President, NMO, Uttarakhand State. Sir has been CME speaker and faculty at 22 scientific conferences. Special courses conducted by him are in medical education, various training, CME workshops of around 42. He has 33 research presentations in various conferences, both national and state. He's been chairperson in 19 scientific sessions in the conferences, and he has attended 36 conferences. He has 11 published books, 83 research publications, and 1 book chapter. Sir has 29 plus years of teaching experience in the medical profession, has been awarded 14 awards, including the 2 Best Teacher Awards at the AIMS Rishikesh. He is the office bearer, elect Anatomical Society of India 14 times. And he was the office bearer at National International Professional Societies, 7 in number. So we welcome you, sir, for the session. And our next chairperson is Dr. Biswa Beena Ray Ma'am, Professor and Head, Department of Anatomy, AIMS, Kalyani. Ma'am is a graduate of Nilratan Sirkar Medical College, Kolkata, and MS from Institute of Medical Sciences, Banaras, Hindu University. She is Associate Editor in Clinical Epidemiology and Global Health, Section Editor in National Journal of Clinical Anatomy. Ma'am has 47 publications in international and national index journals and 22 PubMed index articles. She is reviewed in reputed journals, including Clinical Anatomy, Anatomical Sciences, Education, and European Journal of Anatomy. Welcome you, ma'am, to the session. And now we start the session, and I'm giving it to Dr. Dinesh.
So now the session is yours, Dr. Yogesh sir. I hope this session will be immensely useful to all the participants regarding histological image analysis. We welcome you, sir.
Yes, sir. Thank you. Thanks for inviting the session. I'm sharing the screen. Dinesh sir, is it visible entire screen?
Yes, sir. It is visible, sir.
Okay, sir.
Yes, sir.
So I'm not going to much in the detail of this software, but before that one, this talk I am giving, before that I would like to say, as an organizing secretary, special thanks to Dr. Sharif sir, Dr. Daisy ma'am, Dr. Surjit sir, Dr. Aruna ma'am, Dr. Ramadevi ma'am, Neem sir, Brijen sir, Bishwabina ma'am, and Vasudha ma'am, because of the thing they have shared their knowledge, and they have given time to attend this meet and to give their inputs during this meet. So to enlighten the participants, I am especially thankful to them. I am also especially thankful to all the delegates who have registered, who are attending, and JIPMER administrations for providing the facility to conduct this workshop. I am especially thankful to all of you. Many participants are not able to join due to some technical issues. Still, I can see the participant number is 570 or 580. It has gone till now. At the end of the session also, it is 460, 470, like that. So I am especially thankful to all the participants for registering this workshop. Before going in the detail of this user manual and usage of this software, I want to share a few things actually. One thing should be in mind: if Dr. Vasudha or Dr. Yogesh or somebody else can use it, I am confident that everyone can use this software. This image software is not having any restriction of the usage, particularly when we say that Is it for histology image? Is it for that? It is the image analysis software. It can analyze any image. It can analyze the entire image of the earth. So that much potential this software is having. Only the issue technical expertise is required, but we are not in need of too much technical expertise. If we know basic tools, we can easily use this software. So I am going to teach only the basic tools what we need for histological analysis. Once we start using this one, you can build your own building to n number of the floors. So we will start with the basic things only. See, when we open the software, we can see the user interface. So this is the user interface that we can see here. The number of tools are there. And below that one, this continuously changing X and Y axis and some values are there, coordinates are changing. That is called as the status bar. So this is called user interface. This user interface is little bit different in Mac operating systems and Windows system. But ultimately our use is same, our image is same, and we are going to analyze the same thing. So nothing to worry whether you are using it on the Mac system or you are using it on the Windows system. So in the Mac, this user interface is always associated with these menu commands. Menu commands means file, edit, image, process, analyze, all these things. User interface and this menu commands are fused together in Windows laptops or Windows system, but in the Mac, little bit difference is there. So in the top here, you can see these commands are coming separately. So these are the menu commands. For example, if we click on the file command drop-down menu will come and it can be used in the same way, in the similar fashion. So how to work for this software? Open the software. For Mac OS, you have to right click the software and then select open menu. In the Windows, double click it, it will open directly. So there is a slight difference in opening the software. So for Mac operating system, right click and open the software. For Windows, just double click the software. I hope so. I am going in the little bit slow space because some of the participants are following these instructions right now. Okay, so to open the image, what they have to do, just go to the file menu and select open. So it will select the open to open the image that can be selected by clicking open, and you can select the location, whatever folder you have saved, that image can be opened easily like other Word files. It is just a software which utilizes like Microsoft Windows. All the things are in the same way here also. So my work is to make you aware about the weapons which are available in this software. I am going to make you aware about the basic weapons that are tools. So first tool here, you can see there is a rectangle tool. You may see two screens simultaneously. One is the PowerPoint, that are the steps. And on the another side, there is an image J software which is running here. So this is the image J software. And I have opened one thyroid gland image. I have opened one thyroid gland image here. Okay, so we can open the thyroid gland image and we can work on that image. Any image you can open. As I told, it can analyze even the image of the earth. It can open any image here. So suppose this is the rectangle and this, we can select the rectangle tool and we can draw a rectangle here. What is this, man? What do you mean by this rectangle? It is called ROI, region of interest. We are selecting a particular specific area of this image and we are going to analyze only that area. So that is called as these all are the selection tools. These are called selection tools. So here a lot of selection tools are there. Based on our uses, we can select the tool, what is required for our analysis. So first tool I am saying is the rectangle tool. So I want square tool. So what to do? There is no square tool in this image software. Nothing to worry. Just select the rectangle tool, and while drawing the rectangle, you just press the shift button. You just press the shift button. If you press the shift button, the rectangle will get converted into a square. So one of the participants has asked, sir, how we can follow these steps. So we have prepared and compiled as a user manual. User manual is free of cost, available in Image software website also, but it is very big. So what steps to be followed? So basic steps we have compiled and we have circulated in the groups for your personal use. Later you can sit together and you read and you can follow the same thing here. So this is the rectangle tool. There is another tool also. If we long press this rectangle tool, you will get another option. One is the rectangle, another is the round rectangle, rotated rectangle. Out of that one, I am selecting now the round rectangle. So if you see carefully, the corners of this rectangle are now converted into the round margin. If we double click this one, it will open another pop-up window. By that one, you can increase the width of the rectangle. You just see I am increasing the stroke width of the rectangle. Corner of the rectangle also we can change. We can make it more circular. We can make it reduce. These are the tools. If once you start using, you just keep on working on it, you will find out more and more interesting pathways for the use. Here it is orange color, you make it black, or based on your image suitability, you can change the color. So how we can open that pathway? Just double click the rounded rectangle tool and you will get this pop-up window. And in this pop-up window, you can change the stroke width, diameter of the corner, color, fill color. I am using none. You can just, instead of the border, you can use the fill color to show that this is the area which we want to analyze. So this is a basic thing here. Okay, so these are the things that we are learning now. Okay, so I hope so this first tool, rectangle tool, is familiar with you now. I will move to the another thing. Before that one, we will revise once again in short the set scale. What is exactly do you mean by the set scale? See, the image which is captured by your camera, you are going to analyze. But this software don't know how much the image is enlarged, how much is the distance of the image from your eyes or your camera. So this software don't understand that one. So what we have to do, we have to take the camera, we have to capture one image, simultaneously we have to put some scale and we have to capture from the same distance that scale also. So concept should be clear. With the same settings, same camera, same microscope, we have to capture two images. One is the slide image or any image, and another is the image of the scale or ruler. So that image will be having same magnification and same everything. So this is the image that we have captured using the 10x and this is nothing but a stage micrometer. This stage micrometer are like a slide like things, easily available. As Dr. Sulochana said, it is easily available in the Amazon and very cheaper cost, hardly 300-500 rupees. And once this stage micrometer will last throughout your life if you don't break it. Okay, so this is the stage micrometer. That image we have captured using 10x. Now I want to analyze another images captured by 10x using same camera, same microscope. So I will set the scale. I will give commands to images software. So this much is the magnification. Actually, all images are having pixels, and that pixels, we will convert pixels value into some value into micron. Okay, so pixel value we are going to convert into micron value. That is nothing but the set scale, because image knows only pixels because it is the image. So like that one, for this one analysis, we need to select a straight line tool and just draw one line from start point to the end point and go to analyze and set scale. So in the analyze and set scale, this one pop-up window will come. In this pop-up window, there is 414 pixels. It will analyze and it will show you. It is different for my image. It may be different for your image. Okay, so nothing to worry. It is showing that 404 pixels. And now I know the distance between start and end point is 1 millimeter. 1 millimeter, 1000 micrometer. So I will just put 1000 micrometer. This software has kept unit instead of unit of length. What I'm going to use unit of length is the micron. Simply, I have converted unit into micron. I am not going to analyze the same image. I am going to analyze another images which are captured by same setting by 10x. That's why I will apply this setting, this standardization globally. So global means n number of images which will be scanned after this one. That will have same value. So here you just see scale. It is showing some pixel value per micron and just click OK. So now your software is standardized. Now suppose this is the follicle, one follicle, and I want to just measure the length of this follicle. You just see it is showing 572 micron. So length of the follicle easily can be measured. This is only we want. In the histology analysis, this is the basic step. What is in our mind? How to measure. Actually, most of the anatomy research in our country, we are lagging behind this use of the software. And that's why we are not able to quantify it properly. This use of ocular and stage micrometer in olden days, as Neem sir said, so that was actually tiresome. It will strain your eyes. So this use is very easy. Just take the micrometer scale and now you can measure the diameter like this one, like another diameter. So length it is giving. I will show you how to summarize, how to take out the value. This is just a base to make you aware how to set the scale. Next selection tool is the oval selection tool. This is the second tool, which is the oval tool. And this oval tool can be used like this one. Just select the oval tool and draw the oval shape and it can be used. This is useful for arteries or some other structures, but it may not be useful to find out the perimeter of your follicle. We will see how to find out that perimeter also. But I want exactly the circle. How to draw a circle? While drawing this elliptical tool, I am just pressing long press, continuous press of the shift button, and by pressing continuous shift button, I can draw a circle. If you double click this one, it will give you the size of the brush and other things. We will not go in detail of this one. So this is the oval selection tool. Are you aware now? You can draw a circle or you can draw a oval using this one. Okay, there is another selection tool that is called polygon tool. This is the polygon tool. Next tool, tool number 3. This tool number 3 is the polygon tool. I have to select this polygon tool, and what I will do, I will keep on clicking here. Okay, I am keep on clicking here. 1, 2, 3, 4, 5. I am keep on clicking here. But to complete it, I will just double click. So it will join the beginning point at the end. Now I did not have to go to the beginning point. Just go near the beginning point and double click. It will make a complete shape. So this shape is ready. Now we are going to analyze what is the size of this follicle. So this is the way by which we can analyze the follicle size or the area of the follicle. Then there is another tool that is the, we are not happy. It is angulated. Some part of the follicle here, if you see, some part of the follicle is outside. I am not happy with this one. I want another tool. I want more accuracy. Yes, we can do it. We can have more accuracy. Okay, how to do that more accuracy? I will select the next tool. Next tool is a freehand selection tool. This freehand selection tool is this one. Number 4 tool is the freehand selection tool. I have sent a link. I have sent already the user manual. If you have any doubt, just open the user manual and you can follow the steps. So by this way, you can draw a particular shape which is just encircling. I am not doing it in an exact manner, but this is the way how you can select exactly your follicle. Okay, so exactly follicle you can select and you can analyze the follicle. So this is the way how you can use freehand tool. Line tool, already I have stated that select the line tool and you can draw the line and you can see the diameter of this follicle, or the distance between two cells, distance between two nuclei, distance between one blood vessel to another blood vessel. Thickness, one person was asking, can we measure the thickness? Yes, this is the way. Suppose 10 collagen fibers are arranged in one bundle. Just measures, for example, I feel that this is the one bundle of the collagen fiber. Okay, so I can measure the thickness of the collagen bundle. Even if elastic fibers, they are fine fibers. I have done Warhoff Van Giesen stain or Orsin stain for the elastic fiber. Okay, I will have to zoom it and I will measure a single fiber. In particular field, how many fibers are there, that also we can count. How much is the thickness of each fiber, that also we can count using this software. So this is the beauty of this software. Okay, in the line tool, there is another tool like a freehand tool. There is a freehand line. Freehand line means what I have to do it. You just see, I want to measure the perimeter only. Perimeter means circumference of this follicle, the perimeter of the follicle. Yes, I can draw the line like this one, like this, but it is not the shape. It is just a line. There is a difference between this line tool and freehand shape tool. Freehand shape means area also will come, perimeter will also come, both will come in freehand tool. But freehand line, only the length of the line will come. That is the difference. So I feel so I'm going slowly so that participants can learn and follow the steps. Okay, suppose I have used this line tool, straight line tool, and I have measured the diameters. Mistakenly, I have drawn a long line. I can go to one end of this line, I can hold and I can make it smaller line. Or I can rotate it. I can hold the vortex and I can rotate it. I can hold the entire line and I can shift the position of the line. So these things are also possible in the line tool. Okay, then I feel that there is an angle between one follicle and another follicle. So how much is the angle? Suppose one collagen bundle is running in one direction and another is running in another direction. How much is the angle? So that angle measurement also can be done using the next tool. That is the angle tool. We will see the arrow tool later. So angle tool is also there. Okay, to measure the angle, I am putting one point here, another point here, and another point here. So this is the angle which can be measured using this tool. So after drawing this one, we have to go into analyze and measure. So it will give you the value, one angle we have drawn, and the angle is 125 degree. The angle is 125 degree. Similarly, you can go and draw another angle. Again, you will have to go to analyze and measure. It will add another, it will add another angle in your finding. You just go analyze and measure. This is the step how we can measure the things actually. Result table has gone in the back side actually. So I am putting it, bringing it front. Here are some documents. If it is open, you may not see this result table. This is called as the result table actually. This result table will give angles. I have drawn 3 angles actually. First is 125, second is 121, and another is 115. How much is the mean? How much is the standard deviation? How much is the maximum angle? How much is the minimum angle? That is in our mind. We will copy this one and paste it in Excel sheet. Then we will try to do it. Nothing to worry. Just right click and summarize. You will get all value, mean, standard deviation, minimum, maximum. All values can be shown by this software very nicely and very easily. Follicle, 1 follicle area, 2 follicle area, 3 follicle area. So what is the mean follicle area? What is the standard deviation between the follicular areas? So that can be easily done using this software. I will come to this point once again. They will ask to save this result or not. No, need not have to save this result at the time, or you can select and it can be saved later also. Okay, so we are going to move to the angle tool. I have already explained. Then there is a tool called arrow tool. In this one, there is another tool. In the line, there is an arrow tool. So this is the arrow tool. This arrow tool we can draw and double click on the arrow tool. You will get one pop-up window. In the arrow tool, you can increase the width of the arrow. You can increase the size of the arrow. You can even change the color of the arrow. I want yellow arrow. I don't want filled arrow. I want only the outline, so that the things which are deep to the arrow, that should not be hampered. That should be visible. I want double-headed arrow. Both the right and the left head arrows I can have like this one. So these are the things which we can draw using the arrow tool. So coming to the next tool, that is the point tool. This point tool, there are 2 types of the point tool. Okay. So there are the 2 types of the point tool. Okay. So one is the simple point tool. That means I want to count the cell. This is first cell, this is second cell. Oh my God, what is happening? Can you see? Only one point is clicked, another point. So how to do? Yes, it is very easy. See what I am doing. I am clicking on one point. At a time, one point will be analyzed, but I want to count it. So what I will press the shift button. I will press simply the shift button. First point, second point, third point. You just see now numbers are also coming 1, 2, 3. It may not be clearly visible, but labels of the arrow, this label of this point tool will also come. Double click the point tool, you will get the option also. Here dot hybrid arrow. So I want a little bit bigger, extra large. I want double XL, very big dots I want. So the size of this point also can be changed. Okay, so like that one, you can change the color. Here it is the orange color. You can make it yellow. You can make it black color. So it depends on what you want. So this is the point tool and you can analyze afterwards. So point tool is having two tools now. One is one point tool and and there is another tool called as the multi-point tool. If we long press this point tool, you will get a multi-point tool. And multi-point tool, no need to press shift button. One cell, second cell, third cell, fourth cell, fifth cell, sixth cell, like that. On our follicle number, you can have for one follicle, 2, 3, 4, 5, 6, 7, 8, 9, 10, whatever you want, you can count it very easily. So this is the way how we can use the point tool, single point and multi-point tool we can use. But I want to delete some points actually. So for deleting point, just press the alter button or option button and click on that point. See, other numbers are changing. I don't want this first point. So last point is point number 9. So last point is point number 9. I will press alter and click on first tool. So the number will automatically reduce. It will renumber the remaining points. So alter and click the point so that point will be removed. Okay. So there is another tool called as the WAN tool. There is another tool called as the WAN tool. So this WAN tool, once we click, okay, so nothing is visible, nothing to worry. Just click somewhere in the follicle center and double click this one. And another pop-up window will come. I want to measure the follicular area, colloid area. So what I will do, I will increase the tolerance. It is little bit easy tool actually. See, the follicle is now marked and I can measure the follicle, exactly the colloid material, how much is the colloid material. So to increase the size or the decrease the size of this one, we have to scroll the tolerance bar. There is a tolerance bar that we can scroll and we can increase or decrease. So this is the way how we can use this one. Okay. Then there is another tool, that is the text tool. Already Dr. Sulochana has covered this text tool. I will just revise it fastly. There is a text tool. Just select the text tool and type it. You can type thyroid and you can change the color. Double click it, the text tool. It will allow you to change the font size actually, serif or sans serif, plain, left, align. So size of the font is our main issue. See, this is the size of the font that can be changed. Color also I told we can make it black or whatever we want, like that one we can do, use the text tool. Then there is another tool called magnifying glass. This magnifying glass is there. So select the magnifying glass. It will increase your figure or it will decrease the selection. Okay, so like that one, while clicking, you can press the option button to zoom in and zoom out. So like this one, you have to do it. Okay. So this is the way, plus sign and minus sign, plus sign, minus sign, upward arrow and downward arrow, both will have the same function, like a magnification and demagnification, like that one. See, we are not magnifying the image. We are magnifying just the view, what we are seeing. We are not enhancing the size of the image. Size of the image is something different. It is in pixels or it is in TIFF file. So that we are not changing. We are just zooming in and we are zooming out, like that one. This is the use. Then there is another tool called the scrolling tool. There is another tool called scrolling tool. Sorry, this has
Yogesh sir.
Scrolling tool.
Yogesh,
Yes, sir.
Ma'am has asked to just repeat about WAN tool only, sir.
Okay.
WAN
tool. I am repeating the WAN tool, sir. The WAN tool is useful to select the follicle colloid material. This area I want to select, so it select only a specific area. It selects the color intensity. Actually, it identifies which is the color intensity. So this is the WAN tool I am selecting and I am clicking it. See, this is the already I have done setting. So double click this WAN tool. And if I am satisfied, then it is okay. Or I can increase. See, it is selecting a particular area. Whatever area we need, we can increase or decrease the tolerance. This is a very useful tool. It saves our lot of time. Otherwise, we have to use the freehand tool and we have to circle that zone. It takes a huge time. So this is the better tool, that is the tolerance tool. Tolerance tool will have little bit less accuracy, but it saves our effort to select manually each and every follicle. See, this is one follicle. I will show again in the another follicle. By clicking this one, WAN tool, I can click and I can reduce the tolerance like this one. So I hope so it is useful now. Okay, so scrolling tool is the another tool. Scrolling tool means we have to enhance like this image, then only we can use the scrolling tool like a hand tool. In paintbrush, there is a hand tool. We can scroll the image. Only if the image is too much zoomed. Suppose this follicle is visible, another follicle I want to see. So that time only it is useful. Okay, so to work in the smaller area of the image, this zoom tool is useful, scrolling tool is useful. There is another thing now we will go, that is the contextual menu. If we see, this one is the image. So when we right click the image, right-clicking the image, it will give some option. There are a lot of options. I am not going in detail. Already we have a delayed session. Nothing to worry. Only you can use the rename, duplicate, or so many other controls are there. That contextual tools means right-click and use some options. In the Word file like we are doing, same things here also we can do. Now we go to the another thing, that is the file, edit, image, the menu bar. In the menu bar, already file is covered by Dr. Sulochana and Dr. Gladwin sir. I'm going directly to the edit. We have edit options. So when we go and edit panel, go in the edit panel, there is undo, cut, copy, paste, lot of things. Like a Microsoft Word, here also we can do. For example, I want to draw one rectangle. Here I have drawn this rectangle and Command C, Command V, I have pasted C. This area is copied now. So like that one. Okay, so these are the things that we can do it. Okay. Sometimes while working, you may do something wrong. So you want to go back. So that reset also can be done. There is a Command Shift R button. Suppose, for example, I have selected, I have deleted something and it is now the image is having hole. So how to go back? There is an option, Control Shift R or Command Shift R. It will ask you, do you want to revert the changes? Yes, you can revert the changes like this one and your image will become original image like this one. So undo, cut, and other things like copy, paste, delete. These are the options as the Microsoft Word. We can do it here also. Then there is one option. In the olden days, we used to have that negative, photograph negative. So same thing we can do it here also. That is the Command Shift and I, or we can invert it. You just see here. Okay. So Command Shift I means we can invert and we can make it like a negative. Some option we are having confusion whether it is nucleus or background. So we can revert the colors and we can see it like this one. So I hope so. This change is also useful sometimes to identify more and more things. Okay. So this is the thing. Then we can go to the edit and selection mode. See here, this is the image I have selected and edit and selection. In the selection option, there is two options, select all, select none. Suppose lot of area you have selected and you don't want to select any area. So select none and select all. For example, there are shortcuts. I have selected this area. I don't want to select. Then Control Shift A. So none is selected. and Command A or Control A, entire image is selected. So like that one, these are the options: select none and select all. There are always shortcuts to perform each function. There may be the another shortcut. Now, one important thing I want to say, that is the straighten. Straighten means what? This you people learn. This is very important tool. For example, sorry. Okay, this one I'm reducing little bit the size. See here, this is the follicle. This is the follicle, and I want to see how many number of the nuclei are there in one line of the follicle. So what I will do, I will use freehand line. So I will use the tool freehand line. I am drawing one line here. I am drawing one line here. Okay, and I will double click that one, and it will increase the line width. I will just thicken the line. You just see the line thickness is increasing. The baseline one black line will be there, that will not around surrounding the area that is there. So this line I have created. Then what I will do, I will go to the image, I will go to the selection, and I will go to the straighten option. So one another image is now visible. You just see, this is the another image. This is the another image, and this another image is having all the nuclei in the straight line. 1, 2, 3, 4, 5, 6, 7, 8. So the nuclei which are in the curve, it will create another image. For some analysis, this tool is useful. So that is the straighten tool. Okay, so another tools are also there. That is the round rectangle tool. I am not going in detail. Reset, I have already told reset. There is an option that is called as the Control Shift R. Control Shift R means revert all the changes. And sometimes you have done some changes in this image software. So that changes can be done image, and you have to go to option and reset. So this option is there, reset option. Reset means it will reset your image software. So sometimes we do some selection or something, so that can be reverted using this software. So there is another menu that is called as the image menu. So in the image menu, you can have a lot of other things, but only one thing I will tell. So image, we can have brightness, adjust and brightness. We can adjust the brightness. You just see, we can increase the brightness, we can decrease the brightness. So brightness and contrast can be done, or you can select auto. Then you will have to apply if you want. Okay, so these are the things in the image. Lot of things are there. I'm not going in detail. We can convert it into the black and white image also. Okay, so these are the things that we can do it there. We can change the size of the image also. We can increase the pixel size. We can decrease the pixel size. These are the options in the image also. So another is there, we can have the option called as the in the image. Then we have to go to transform, and we can flip the image. We can flip horizontally, we can flip vertically, we can flip. So that flipping is there. Flipping in the particular axis is also there. So these are the things that we can do it. Even we can zoom. So zoom option also I told, plus, minus. These are the options by which you can zoom in or zoom out the image. So that have two options, either use plus minus sign, or you can go to image, select the zoom, and then select whether zoom in or zoom out. So why to go in this long process? But here are two options. One is short key, another is the long process. You can follow both, whatever you feel good. Then there is another option called as the overlay. What is overlay? For example, I am drawing one rectangle here. Okay, so one rectangle I am drawing here. I want to draw another rectangle. See, not possible. See, not possible. So there is one option. One rectangle once I draw, then go to the image and select lot of shortcut I can tell, that is the Control B or Command B. You just press Command B. Now you just see you can add another rectangle. Command B, you can add another rectangle. Command B, you can add another rectangle. Command B, you can add even another circle. Command B, you can add your any other tool also, like that. So these are the things that is called as the overlay. Command B means overlay. You just see, if I zoom it, I am zooming it, the line thickness of this overlay is not changing because I have not disturbed the image. But once I import this image, it will not be there. It will disappear. I want one image which should have these shapes as it is. So for that one, I have to press Shift and F to flatten. See, new image appeared, and now this new image is saved in same folder with the another name, that is -1. This image is not having -1. This image is saved as thyroid gland -1. So this is the difference, and now it is flat, and this we can send it for publication or some other purposes we can use for the print media. Once we zoom this image, now this is blurring. Actually, these corners are blurring because these are embossed now in the images. So this is the thing that we have to work. Okay. So this is called overlay. Now we have to see the ROI manager, the most important two things. One is the set scale and another is the ROI manager. If you don't learn these tools are very easy, that I have shown, you can learn it easily. But ROI manager, measure, set scale, these are the three basic things that are important. What is ROI manager actually? Suppose for example, I have drawn one circle here. This is one circle. This is suppose the follicle, and I will overlay, Command B or Control B. Then I will go to the image, and from the image I will go to overlay, and in the overlay you will get to ROI manager. To ROI manager is very important. Understand where it is. It is in the image, overlay, and to ROI manager. This is prime important step of image J. Now this is one circle that I have drawn, and now this is the circle, but I don't understand this name. After 10 days, if I want to see what is this, I don't understand this number because all results will show this number. So I can rename it. I am just renaming it. That is the circle 1. Okay, whenever I am getting result, that time it will show the result of circle 1. Now I am going to draw one rectangle here. See, one rectangle, I am drawing it here, and you just see here, and I have to add it. I have added the rectangle. It has given some another name. So I will rename, and I will put the rectangle name, rectangle 1 to it. So now two things are there. I will draw another polygon also now. So polygon is there. So I am going to draw one polygon, 1, 2, 3, 4, and 5 something. I have clicked one polygon. Then I am going and adding it. Okay, sorry. Okay, so I have created one polygon and I can add this polygon. Again, new number came. So what I will do, I will rename it as a polygon. Okay, so this is the thing actually. So these are the things: circle, rectangle, and polygon. 1, 2, and 3. Very, very important step. Just go and click measure. You are getting entire data now. Your entire data, this only we want now. What is the area of the follicle? What is the length? What is the size? That is there. So this is circle 1, rectangle 1, polygon, and these are the areas. These are the perimeters. Now, I want to average perimeter. I want a mean area of this one. Right click. This is the result table. Right click on the result table and summarize. You will get the mean area. You can get the standard deviation of the area. That's all regarding this software. This is the prime important thing. Few things are there. Same things we can have in different way. But only few things I want to add in this one. I will revert this image table. If you want to save the table, you can save. You just select the save. It will ask you the location and other things. Otherwise, it will get saved in your file. Save. Okay. It will get saved in your same folder, and you have to name it. Okay, so I am processing now. I am reverting the changes. Okay. Command Shift R. So this is the image now, and we have some other options also. So image, we can smoothen something. For example, I am selecting one particular area and I don't want this much area to have very sharpness. So I can go and process and we can smoothen it. So it will become hazy, more hazy, like that one we can do it. Okay, so we can make the changes, undo also, Command Shift R, and our image will become original one. Even we can go in the process and we can sharpen. It will sharpen and it will make crisp clear to the nuclei. Like that also we can process the image. And this is the analysis already I have told, and summary table also I have told. Final step is how to add, don't save, I will reopen the image, file, open recent, and we can recently open thyroid gland. So this is the image. In many journals we see some bar is there, that is called as the scale bar. This magnification, because journal file printing it, they may compress, they may enlarge, so they don't know anything about this image. So scale bar is the standard thing that you have to add in the image at the end. So how you can add? You go to the analyze and you use the tool scale bar, and then one option will open here. See, this is the pop-up window and this is 257 micrometer. So I want only 100 micrometer scale bar, or I want 500 micrometer scale bar. So here in the bottom of the image, one scale bar is there now. I don't want it in the white, I want it in the black color, that is there. Then font size too much, 500 micrometer is too much small there. We can increase the font size to 30 or something, whatever we want, and it will be printed. There are so many other options. Font also we can change font size, everything. So now your image is having this scale bar, and you just go to the file and save image, save or save as. Save as TIFF, this image will be saved as it is. I feel so. I have tried to cover, as we are lagging in the time. I have tried to rush. These are my books for which mostly you people knows me. That's all about this session. Jai Hind. Thank you. Dinesh sir, it is over to you, sir.
Thank you for the wonderful session, sir. Now I request the chairperson, Dr. Brijendra Singh sir, to give his valuable opinion and questions, if any. Dr. Brijendra Singh sir, please.
Right.
Sir. Ma'am, okay. Dr. Biswabina, ma'am, you can give your valuable suggestions and feedback.
Thank you. Am I audible?
Yes, ma'am, it is audible.
Okay, thank you very much for the organizers. I never imagined that an online workshop can be so informative. And actually, so in my life, the best teachers, whenever I have listened to their classes, I thought, why not 1 more hour? So that is the success of this workshop. And then secondly, about the speaker, Dr. Yogesh Sontake is a known personality in all over India and internationally, of course. So a person with very clear concepts actually can convey so clearly. His books are also rich with all clear concepts, but here his way of explanation shows me how much deeply he has understood the concepts, and then he has conveyed in a very clear, I mean, a very good language. Only thing is that I would have loved to hear for another maybe 3 to 4 hours, even more in more detail. About asking some questions, one thing which clicked my mind is, can we use the same software for live cell imaging?
Yes, ma'am. This is also useful for live cell imaging. For example, if we see there is one image I have opened. This is the image which we have taken it actually, and in this image we can use it multiple point tool. In the olden days, this I will have to share the screen. 1 second, sir. I'm sharing the screen. This is the image which is Dinesh sir, is it visible? Okay, so this is the image and multiple point tool which we can use, and live cells we can number it like that one, and we can go analyze and measure, and it will give you the values, how many cells. So 4 cells we have counted. So there are 2 aspects in the live cell imaging actually. 2 types of culture will be there. One is the monolayer culture. So monolayer culture counting is very easy. Z stacking is not required. So stacks are not there. And in the multi-layered cell culture, actually multiple stacking will be there. For that one, you need the live cell imaging microscope, which will stack the cells actually in the different zones, and that can be similarly counted and measured it. In the olden days, this image J was having separate cell counter, but now they have introduced this multiple point tool and they have resolved that issue. So once if somebody wants to listen this one in internet, in YouTube, they may see that cell counter videos. But nowadays, I feel that are not visible. It is very easy, multiple point tool, and Z stacking will be there if it is multiple layer, and in the single layer, monolayer, it is very, very easy. Like nuclei, we can count like that one. Live cells also we can count. I hope so. This is answer.
So multiple images have to be used for counting the cells in a case of a live cell counter.
Yes,
and
yes, multiple images will be required.
Okay, my second question is there any applicability of image software in gross anatomy research?
Yes, I feel so. This also can be used for gross anatomy images, but we need to put some scale actually. That is only one drawback while using the gross anatomy images. For example, heart image you are taking. So while taking the gross anatomy image, we need to put some ruler or something there simultaneously. So, and that ruler with the same image can be utilized for standardizing or setting the scale or calibration of image J. But it can be easily used to measure the angle. For example, left coronary artery is dividing into the left circumflex and left anterior descending. So the angle between that can be easily measured using this software. No need to use any other things and dividers and compass. So it is very easy. And proportional analysis. The proportional analysis means proportion of the thickness of one blood vessel with the another blood vessel. That can be easily used using this one.
Okay, thank you very much. And thanks for inviting me for this informative workshop. I'm really interested. Thanks a lot.
No, thank you. We are thankful to you, ma'am, that you have joined and you have given your valuable time and you have enlightened the event. Thank you. Thank you so much.
It was worth it.
Thank you. Thank you very much, ma'am. Now, I wish to ask certain questions which has been raised by the audience. First thing is, what is the question of reliability related to the image J? Is there any specific times we need to calculate so that we can achieve the reliability, or is the reliability an inbuilt one in the image J software?
No, see, this is, it is not doing anything. It is just giving the results and output. What input we are giving, it is giving same output to us. So standardization depends on us. How much accurately we are standardizing this, we are calibrating it. It is based on that one. So it is 100% reliable because it is not doing anything. It is just measuring the pixel and converting it into micron and giving us some value. So it depends on us only how much input we are giving. So inputs based output it can give.
How to undo previous sections which we have used, previous selections?
Previous selections, there are two things we can do. We can just completely revert all the changes. To completely revert all the changes is Control Shift A or Command Shift A. So by that one, you will unselect all selections. This is one thing. Second is reverting all the changes. Reverting means the image will go back to our original image. So Command Shift R for revert. So Command Shift R or Control Shift R is for reverting all the changes in this one.
Quantify the intensity of the cells in the images.
No, what you are saying, sir?
How to quantify the intensity of the cells in the image? I think so. They have asked related to IHC, sir.
Yes, that stereology and other aspects I have not covered. It will take a long time, sir. We will arrange special session.
Yes, sir. Then one more question is, how do we differentiate perimeter and length in sections, because both are giving the same value in image? Perimeter and length.
Yes, sir. There are two tools. One tool is rectangular tool. So rectangle means it will give complete perimeter of the rectangle. And for length, measuring the length, you have to measure only using the line tool. There is a line tool, that one. And if you are using rectangle, then also it will give three values. One value for the area, another four value it will give. Area it will give perimeter, it will give length, and it will give width. So four values it will give. So it depends what you want to do. It may show n number of values. Out of that one, what are useful, that we have to take out.
The question is how to reach the scale bar and use it, sir, because some cannot get it, it seems.
Okay.
So if possible.
Yes. For scale bar or standardization or calibration, first requirement, you need stage micrometer, which is easily available Amazon or other websites. Second thing, you need camera fitted microscope. Okay, so that is second requirement. In 10x, you take the image. So that are the basic. You should have one image of 10x of stage micrometer. And after taking that one, then you have to go to the certain steps, that is the analyze and set scale. Open the image. After opening the image, you have to draw one line, that is the point 1 to last point, that is the 1000 micrometer length line, and then go to the analyze and set scale. After clicking that one, you will get option, and in that one, rather than going in detail, please refer the manual which is provided. Okay, it will, yes, I think so, it will solve the issue.
Yes, sir. Then one suggestion is related to which microscope is better for usage for capturing the images and analysis.
Option should be diplomatic. By sitting in government setup, I should say all microscopes are good.
Yes, sir. Then related to this, sir, how far we can retain the originality of the image if we are using an image?
No, I'm not getting question, sir.
How to maintain the originality of the images if we are using image for gross anatomy, like that they have asked, sir.
Sir, images, it is not going to alter anything. See, increasing brightness and contrast, I don't think so it will affect gross anatomy images. Like that one, we are using more bright light, daytime light. So like that one, we are not distorting any image here. There is no option to distort the image. That distortion option you will get in Photoshop.
Yes, sir.
Okay, so distortion options are not there. So 100% originality is maintained in this software. It is not distorting anything.
Yes, sir. So with this, we will be ending off the question and answer sessions. Remaining sessions, we can take it off. You can put in the chat box. We will take the questions and we can answer it and we can send off to you. And so with this, we are concluding the session. And we heard that Dr. Brijendra Singh sir has another one meeting and is currently attending it. So we can. Okay. Now I ask the chairperson, Dr. Biswabina Ray ma'am, to unveil the certificate for Dr. Yogesh sir. Ma'am.
Yes, Dr. Yogesh, thank you very much for the great presentation, and the certificate is all yours.
Thank you. Thank you, ma'am.
Yes, ma'am. Now I ask Dr. Suma ma'am to unveil the certificate to the chairperson, Dr. Biswabina Ray ma'am.
Thank you, Dr. Biswabina Ray ma'am, for spending your time with us and giving your valuable suggestions and comments. Thank you very much, madam.
Yes. With this, we are coming to the end of the scientific sessions, and now I invite the last session for the day is summarizing the workshop, and it will be done by Dr. Dharmaraj Vamanrao Tamgire, Associate Professor, Department of Anatomy, JIPPER. Dr. Dharmaraj Vamanrao Tamgire is currently the Associate Professor in Department of Anatomy, Academic Center, JIPMER, and he has the educational qualification of MBBS and MD Anatomy. He is known for his humble approach and he is a very profound in gross anatomy and other areas. So I invite Dr. Dharmaraj Tamgire sir to give his concluding remarks and summarize the workshop. Welcome, sir.
Thank you, Dr. Dinesh. Am I audible to all? Is it?
Yes, sir, it is audible.
Okay, right. So I don't have any presentation. I will be reading out and summarizing the workshop. Okay, so good afternoon to one and all. It has been a very well spent morning today with wonderful and very informative sessions delivered by eminent speakers today on the eve of National CME on micrometry in microscopic image analysis using ImageJ software. Awareness of such software is an added virtue to our CV and opens up new research avenues. More so, this software being freely available. The immense enthusiasm and curiosity of the participants was evident by the number of registrations which nearly crossed 1,000 for this CME. And I'm confident that we have stood up to your expectations. We were blessed to have eminent speakers in the field with practical hands-on experience on the software, which have given us a greater depth of uses and applications of ImageJ software. Wherein Dr. Suma Madam, Professor and HOD Department of Anatomy, JIPMER, Puducherry, have given the glimpses of software. Dr. Vasudha Kulkarni, Associate Professor, Department of Anatomy, Dr. B.R. Ambedkar Medical College, Bengaluru, has discussed the role of ImageJ software in medical research. Installation of any software is a tedious process and may involve many troubleshoots. Dr. Victor Gladwin has demonstrated efficiently the installation process in a very lucid language and the system requirements for the software. Understanding of user interface is an integral part of any software application, and good understanding of it is an essential prerequisite for the use of software. And I think Dr. Sulochana, Additional Professor, Department of Anatomy, JIPMER, Puducherry, has done a great justice in explaining the user interface of the software. At last, Dr. Yogesh Sontakke, Additional Professor, Department of Anatomy, JIPMER, Puducherry, has explained in detail the actual image analysis and various steps involved in it over the ImageJ software. In addition to this, you are also being provided with a booklet which describes all these processes in detail, which is also compiled by Dr. Yogesh. The current CME is a brainchild of Dr. Yogesh, and second such workshop in line and many more to come. Because of COVID restrictions, we had to conduct it in online mode, but doing so, we could reach to a maximum number of participants. Conducting such CME requires support and coordination from every level of hierarchy. First of all, on behalf of Department of Anatomy, I would like to thank Dr. Rakesh Agrawal, Director, JIPMER, Dr. Pankaj Kundra, Dean Academics, JIPMER, Dr. Dorai Rajan, Chief Medical Superintendent, JIPMER, Dr. Thapa, Dean Research, JIPMER, Pondicherry, for their continuous motivation and support for conducting such CMEs, and appreciate all the support extended for this activity. I would also like to thank Dr. A. Sharif, Professor and Head, Department of Anatomy, AIMS, Delhi, Dr. Daisy Sahani, Professor and Head, Department of Anatomy, Postgraduate Institute of Medical Education and Research, Chandigarh, Dr. Surjit Ghatak, Professor and Head, Department of Anatomy, AIMS, Jodhpur, Dr. S. Aruna, Professor and Head, Department of Anatomy, IGMC RI, Puducherry, Dr. Rema Devi, Professor and Head, Department of Anatomy, TIMSS Puducherry, Dr. V.K. Neem, Professor, Department of Anatomy, TIMSS Puducherry, Dr. Brijendra Singh, Professor and Head, Department of Anatomy, AIIMS Rishikesh, Dr. Biswabina Ray, Professor and Head, Department of Anatomy, AIIMS Kalyani, for consenting and sparing their precious time to chair a session in today's CME. Thank you all. The only external speaker for today's CME, Dr. Vasudha Kulkarni, Associate Professor, Associate Professor, Department of Anatomy in B.R. Ambedkar Medical College, Bengaluru, who discussed micrometry and its application in medical research. We thank you, madam, for your contribution. Dr. Suma H.Y., Professor and Head, Department of Anatomy, JIPMER Puducherry, is always encouraging for such activities. I thank her for inspiring involvement during the conduct of this workshop and also for delivering a session to the CME and being the organizing chairman for this activity. Thank you, madam. I thank Dr. K. Arvindan, Professor, Department of Anatomy, Dr. Sarasu J, Professor, Department of Anatomy, JIPMER Puducherry, for the encouragement and support during the conduct of this CME. I also thank Dr. Gladwin for sparing his time to deliver a session in CME. I would like to thank Dr. Ravindranath, Additional Professor, Department of Anatomy, JIPMER, for his support and encouragement. I will also thank Dr. Sulochana, Additional Professor, Department of Anatomy, JIPMER, for delivering a session and extending her support to the CME. I will thank Dr. Suman Verma, Additional Professor, Department of Anatomy, JIPMER, for being the coordinator for the CME and extending her support to CME. I will thank Dr. Raj Shekhar, Additional Professor, Department of Anatomy, JIPMER Pondicherry, for his support and encouragement. The last in the line, the last in the list, but the main man behind today's CME and organizing secretary for today's CME, Dr. Yogesh Sontake, Additional Professor, Department of Anatomy, has taken every effort to make this event a success. It was possible only because of his continuous effort. I think we all will join me to thank him for organizing such CME and delivering the main content in the CME. Thank you, Dr. Yogesh. It is always a pleasant experience to work with Dr. Dinesh, Associate Professor, Department of Anatomy, Puducherry. He has coordinated the entire program very efficiently, and it is my duty to thank him. Thank you, Dr. Dinesh. We have a dedicated team of senior and junior residents who are our support for the groundwork. I would like to thank our senior resident, Dr. Arun, Dr. Chaitanya, and Dr. Zahira. And the team of junior residents, which includes Dr. Sivasakti, Dr. Abhirami, Dr. Regina, Dr. Sandy, Dr. Urvi, Dr. Grace, Dr. Nandini, Dr. Kalaivani, Dr. Akhil, Dr. Ambika, Dr. Niklesh, Dr. Ambili, Dr. Jayashri, and Dr. Udit, for their active participation for the conduct of this CME. I would also like to thank staff of IT Wing JIPMER for extending the technical support for today's program. I will fail in my duty if I do not mention the administrative and the support staff from the Department of Anatomy for extending their help to the CME. I thank everyone else who, if by mistake, has escaped the above list. Thank you all. Over to you, Dinesh.
Now we have Dr. Brijendra Singh sir with us, and we would like to have few words from Brijendra Singh sir regarding the session and regarding the entire CME as well. Please, sir.
Thank you very much, and this for opportunity to be here with you and to listen this wonderful talk. I have been listening since morning to all the sessions almost, but I am really sorry that due to certain urgent situation, I was called to Director and President's office, so I missed that few period. This workshop come CME has been really a great pleasure, because tool which should be used, how it should be used, and specifically all our PGs, young researchers, and even old and middle aged people like me, who they have learned really a lot. This has been really extremely wonderful. Lot of knowledge has been, practical knowledge I can say, has been shared. How to measure the different things, how to, you can say, okay, not only the measure, how to draw the tools, use the tools, and it's very practical and effective. And I think that this type of CME or workshop on a larger scale must be conducted somewhere else in the near future. That will be a right way to say a big thank you to all of you.
Thank you, sir. So now I ask Dr. Suma ma'am to unveil the certificate for Dr. Brijinder Singh, sir.
Thank you, Dr. Brijinder Singh, for your presence in the workshop and giving your comments. Thank you for sparing the time, sir. Thank you very much.
Thank you, ma'am. Thank you very much.
Thank you, sir.
Yes, sir. Thank you, sir. So with this, we are coming to the conclusion of the workshop, and indeed, it is indeed my great pleasure to be the master of ceremony for this workshop. And I thank Dr. Yogesh and Dr. Suma ma'am for giving me this opportunity and entrusting such a big responsibility, because to conduct a ceremony, we have to coordinate multiple ends at the same time, and there were a lot of practical glitches in arranging and coordinating multiple ends. But still, we believe that we have rendered our maximum level best because of the help of our team of senior residents and junior residents, and without whom we could not have arranged such a session of a big caliber. And entertaining many participants at the same time would have never been possible without an online platform. The initial strength which we were planning for the offline platform was nearly 30 to 40, and with the restrictions and travel, we could have got the participants only from Pondicherry and from Tamil Nadu. But with assuming the online presentation, it was indeed a blessing in disguise, and we could get more number, like the attending participants number itself, it was nearly 550 and 580 at many times. So that itself is an indicator of the success of the program. So we hope to have an offline hands-on session in the near future, and we are extremely grateful to the organizing team members, to the entire set of residents for coordinating and conducting such a big workshop. Thank you all, and thank you for attending the session with patience. And we once again thank you for attending and participating in this session. Thank you. So now we can log off the session, and we are officially coming to the end of the CME. Thank you.
Thank you. Thank you so much.
Thank you, sir. So now I wish to give the certificates. I asked Dr. Suma ma'am to unveil the certificates to the set of residents who have been working day and night for organizing the session and who have been coordinating all the ends. First, Dr. Jahira Banu, Senior Resident, Department of Anatomy. Okay. Now I ask Dr. Yogesh sir to give the certificates to the set of residents. Dr. Jahira Banu T, Senior Resident, Department of Anatomy, JIPMER. Okay, Yogesh sir, left, okay. Dharmaraj sir. Okay, Yogesh sir. Yogesh sir?
Yes, sir. I am hearing you.
No, yes, sir. Yes, sir. Now, I wish to give the certificate of appreciation to Dr. Sivasakti M, Junior Resident, Department of Anatomy, JIPMER.
I am thankful to Dr. Shivashakti and entire senior and junior residents and the supporting staff, Denmark, Chinnaswamy, Neeraj, and all for supporting us continuously day and night. I can't express my feeling towards my residents, how they have worked and they have supported. I am especially thankful to Dr. Suma ma'am for encouraging and supporting continuously. N number of times I have called, she has replied, she has responded. Dr. Dinesh sir, Dr. Dharmaraj sir, I am really thankful for the wonderful and a lot of efforts. And Dr. Suman madam, she has taken the charge and she has coordinated the event very nicely. I am thankful to Dr. Suman and Dr. Sulochna. She has supported me like anything. Dr. Gladwin sir, I feel that whenever administration comes, Dr. Gladwin sir is our backbone and continuously supporting us. Thank you all. Thank you all. A lot of people, I am not taking name, but I am really thankful to everyone. Thank you.
Thank you. Next, Dr. Abhirami GR, Junior Resident, Department of Anatomy, JIPMER. Dr. Sandy S, Junior Resident, Department of Anatomy, JIPMER.
Thank you, sir.
Dr. Urvi Sharma, Junior Resident, Department of Anatomy, JIPMER. Dr. Grace Suganya, Department of Anatomy, JIPMER.
Thank you, sir.
Dr. Kalaivani K, Junior Resident, Department of Anatomy, JIPMER.
Thank you, sir.
Dr. Jakula Akil, Junior Resident, Department of Anatomy, JIPMER.
Thank you, sir. Thank you, sir. This boy has worked day and night, I think so. Has he slept? I don't know.
Yes, sir. Dr. Ambiga R, Junior Resident, Department of Anatomy, JIPMER.
Thank you, sir.
Dr. Nikhilesh S, Junior Resident, Department of Anatomy, JIPMER.
Thank you, sir. Thank you, sir.
Dr. Ambili TP, Junior Resident, Department of Anatomy, JIPMER.
Thank you, sir.
Dr. Udit Narayan, Junior Resident, Department of Anatomy, JIPMER. Okay.
Yogesh.
Now I wish to give a certificate formally to Dr. Dharmaraj sir.
Okay, my certificate.
Yes, sir. Because this is our team. Now a certificate of appreciation to Dr. Dharmaraj Tamgire sir for his concluding remarks and being for organizing the workshop.
Thank you. Thank you.
Yes,
Dr. Dharmaraj, sometimes I feel that am I separable from him? Or from my PG life, he just stuck to me. And
itself,
still the bond is there, and I feel so. The bond will be throughout the life.
Yes,
Thank
sir.
you.
Yes, sir. Yes, sir. Now yes,
Dinesh, Yogesh. Dr. Yogesh.
Yes, sir.
Dr. Yogesh, there is a certificate for Dr. Dinesh also.
Yes, sir. Now,
Yes, sir.
we'll give the certificate now to Madam, sir.
Yes.
Yes, sir. Now, certificate of appreciation to Dr. Suma HY ma'am for being the organizing chairman for the online national CME. And I hope so, Madam is left. Okay. Now, the certificate of appreciation to the man who has planned the entire CME and who has conducted it very, very successfully. Like right from the day of registration, we have unveiled the Google form, and after that, like we have been seeing hour over hour, like what is the registration. And I think that like within that particular day itself, the registration has crossed the 300 initially, then 500, then 800, then 1000. So it was like a visible sign of success at the time of registration itself. So, and once again, I applaud Dr. Yogesh Andrage sir for being the organizing secretary in this online national CME.
Thank you.
Thank you, sir. Nice, sir. Next. Certificate of appreciation to Dr. Suman Verma ma'am for being the CME coordinator in the online national CME. Yes.
No, she is our supporting backbone, Dr. Suman,
Yes,
Dr. Sulochna. I am really thankful. Yes.
Yes.
Thank you.
Yes, sir. Thank you, sir.
Suman, you have coordinated everything very nicely. Thank you.
Thank you, sir. Nothing much, sir. You have done more.
No. You have done
for
a lot
Dinesh.
of job.
Yes, sir. Thank you very much, sir.
One thing I want to say, more thing. Dr. Dinesh and Dr. Dharmaraj, they are not just behind this workshop. Whatever I say, they don't say no. Yes, sir, we can go ahead. We can do this. We can do that. N number of suggestions and a lot of support. I am thankful to
Yes,
them.
sir. Thank you very much, sir. Because it is like one idea, and it has been transformed from that level to this level, sir. Right from the day you have discussed, like, okay, we are going online until this moment of the day. There are multiple things which you have decided upon right from the PowerPoint, what we have to bring and everything, sir. So it is indeed a great pleasure for me to do this, sir. Thank you, sir. Yes, sir. Okay. we have handed over to the residents. And thank you all for participating.
Thank you all. Thank you.
So I declare that this is the end of the session. Thank